7.35 to 7.45. So, in general, blood temperature is slightly higher than the body temperature and is 100.4 degree Fahrenheit. What is the normal in healthy man? What is the normal volume? How much blood we have in our body? 5 to 6 liters. Okay. In general, in a healthy man, the total blood volume in the whole body is 5 to 6 liter, and in each minute we are passing 5 almost 5 liter bloods in each minute. Almost 5 liter blood is circulating or pumping through the heart. We learned yesterday cardiac output. And how much per beat is going?
Around 70 to 100.
Yeah, 70. In general, normal is 70. Good.
75,
And
70.
yeah, so per stroke is going 70 ml per blood. And in 1 minute, if the heart's normal beating 75 minutes per beats per minute, then is circulating in 1 minute almost 5.2 liters blood. So that means whole blood is filtering in each minute. Another question, blood makes up to 8% of the body weight. So 92% is from the other organs, systems, everything. But blood makes up to 80% of the body weight. So now we are coming to the different composition. Blood plasma. In plasma, only plasma, not the total blood. Plasma, 90% is water. In that water, lot of things has been dissolved. What are those? Nutrients. We call salts, called the electrolytes like the sodium, potassium, magnesium, calcium, zinc, whatever maybe. There are a lot of different salts we need for our body function. We have nutrients. We have the respiratory gases. What are the respiratory gases is in the plasma? What respiratory gases we have in the blood plasma?
Oxygen and carbon dioxide.
Very good, very good. Then hormones. We have learned that hormones are carried out by the blood. Then we have the plasma protein. Itself is a protein, and also the waste products, because blood is carrying the nutrients and the waste products and also the respiratory gases. Then we have the salt and electrolytes. Without that, we are not going to survive. So, what are the plasma proteins? The plasma proteins are the most abundant solutes in the plasma, most majority of them. These plasma proteins are made by the liver. Plasma proteins are made by liver. What are the different types of plasma proteins? Albumin, antibodies. We have learned the antibodies. We have clotting proteins. So albumin is to maintain the osmotic pressure. Okay. So the blood pressure, osmotic pressure. And then clotting proteins help to stem blood loss when a blood vessel is injured. So clotting proteins help in the Platelet is a different one. It's the platelet is a different cells which take active parts in the clotting. But for the clotting process, the platelet needs the clotting proteins. And in plasma, we have the different clotting proteins. We are called the factors. And then we have the antibodies, which is produced from the beta and beta lymphocytes to pathogens, protect against pathogens. What is the term? Acidosis means when the bloods become too acidic is called the acidosis, and alkalosis is blood become too basic. So what happened in each scenario? The respiratory system and kidneys help restore blood pH to normal, because respiratory exchange is going to eliminate carbon dioxide and get more oxygen, so there will be balance between the acid bloods, and at the same time kidney is going to excrete out as per the need. So now we have learned the plasma. We have the formed elements. Formed elements has made up three components: erythrocytes, leukocytes, and platelets. Platelets also came called like the erythrocytes called red blood cells or RBCs, leukocytes called white blood cells or WBCs, platelets called cell fragments. What is the erythrocytes? These are the red blood cells. Main function is to carry oxygen. What is it looks like? Shape of the erythrocyte is biconcave discs, essentially bag of hemoglobin. There are hemoglobins is inside the erythrocytes. It's mainly the fully loaded with hemoglobin. Anucleate. Erythrocytes is the only cells in the body which doesn't have a nucleus, because for the loading of the hemoglobin, they push back the nucleus. That's only the reason WBCs can live only 120 days, because they are without the nucleus. So when the nucleus has been pushed away from the RBC, it's matured RBC, but that was ready for doing the functions of the oxygen carrying capacity or the carbon dioxide, but it is no nucleus, so it cannot survive more than 120 days. Contains very few organelles. We have 5 million RBCs per cubic millimeter of blood. So you can see all these are RBCs. This is the stain. So this is the RBC or erythrocyte. It's the biconcave disc-like structure. This only can you see, and then you are going to see different, like the small one is platelets, and we have different types of leukocytes, lymphocytes, neutrophils, and more. So what is the hemoglobin? Hemoglobin is an iron containing protein. It contains iron. That's why when there is the iron deficiency, also you can see the low hemoglobin and all, there is a respiratory stress, because it cannot bind the with the hemoglobin, low iron. So hemoglobin is not saturated. Without the iron, hemoglobin cannot be bind with the iron and become able to get the oxygenation. So bind strongly but reversibly to oxygen. What is reversibly means? It can bind and leave depending upon the situation. Each hemoglobin molecule has 4 oxygen binding sites. Important. Each hemoglobin molecule has 4 oxygen binding sites. Each erythrocyte has 250 million hemoglobin molecules. Can you imagine? 250 million hemoglobin molecules in each erythrocyte. That means how many oxygen each erythrocyte takes and transport. Normal blood contains 12 to 18 gram of hemoglobin per 100 ml of blood. This is important to remember the normal blood of an healthy person per 100 ml of blood. 12 to 18 gram of hemoglobin. Now, which can cause the homeostatic imbalance? Anemia. Anemia can cause decreased oxygen carrying ability of the blood. Anemia can be due to the less production of erythrocytes. It can be low hemoglobin. It can be low iron. So it can cause the anemia. Mainly, ultimately, is the erythrocytes. So it's decreased the oxygen carrying ability of the blood. So the patient is going feeling weak, respiratory stress. Then we have the sickle cell anemia. Our RBC shape is biconcave, but sickle cell anemia is the shape is a crescent moon. So it's abnormally shaped. It's called the sickle cell. The RBC looks like a sickle cell or crescent. So that's why it's called the abspas shaped. We have the sickle cell anemia. Sickle cell anemia is very prevalent in African countries. And we have polycythemia, when there is excessive or abnormal increase in the number of erythrocytes. That is called the polycythemia vera. Sometimes who has the polycythemia vera, they have to take out the blood from the body to reduce the high number of erythrocytes. So this is the normal RBC. It looks like biconcave. Both sides are up, down depression. Sickle cell anemia is like the half moon Christian shaped. This is the sickle cell anemia. See, is when we see the structure of this one normal RBC amino acids, how is made up? Only one change. This is glue is glucose and val is valine. Only one chain in the sixth position makes it sickle cell. So only one amino acid, not even a protein, only one amino acid, maybe 146 amino acid composed of this protein. Okay, the one protein is composed of by 146 amino acids both. Only one change in the sixth position. Glucose has been changed through mutation to valine, and then it has been changed its properties. It became the sickle cell. So polycythemia, we are already told, the disorder resulting from excessive or abnormal increase of RBCs. It can be caused due to bone marrow cancer, unwilling division of erythrocytes. Maybe a response to a life higher altitude. Sometimes CRP is called the secondary. What is the secondary polycythemia? Another question. Because who live in the high altitude, they need, because there is a less oxygen in the as you go to the more higher altitude. So that time they may need to produce more RBCs to combat the distress of oxygen. It's called the hypoxia, less oxygen. Increased RBC slows blood flow and increases blood viscosity. Obviously, it's become more thick, dense, or viscous. The blood flows slowly. That's why I told you sometimes who has really high polycythemia, they have to take out the blood periodically. Next is the form elements. We have the WBCs and platelets. So leukocytes is the white blood cells or WBCs. As we know, is a crucial in the body defense against diseases. These are the complete cells. These are nucleus and cellular organelles. RBC is anucleate. There is no nucleus. We learned yesterday that these white cells can move in and out of the blood vessels. It's called the diapedesis as per the need of the site of infection. They can slick out and slick in through the capillaries wall. It's called the diapedesis. This is important. And it can move by amoeboid motion. Amoeboid motion means slowly they extend like called pseudopodia, like a foot, and they crawl. It's called the amoeboid motion. It can respond to chemicals released by damaged tissue. Any damaged tissue, it can release the sense it. We have 4,800 to 10,800 WBC per cubic millimeter of blood. So what is abnormal numbers of leukocytes? Leukocytosis means generation. When the WBC count is above the 11,000 is called the leukocytosis. Osis means generation. So more WBC, it indicates an infection. If you have any infection and you do the blood test, you can see high WBC count. Also, you can get the cancer also. That is unruling. It's a really high. So, but in general, you can see that because why? When there is an infection, they will increase the number because these WBCs are going to fight for the body defense. So that's why the number increases. So it said high WBC count above 11,000 leukocytes per millimeter square means there may be an infection. Leukopenia. Penia means low, abnormally low level of leukocyte. Sometimes it's caused by some drugs like corticosteroids and anti-cancer agents. What is leukemia? Leukemia, as I told you, the WBC is unruling division of WBCs and produce an excess, too much excess WBCs. And generally, when the bone marrow become cancer, then you can see the unruling division and no control of the blood cell production. That is the leukemia. What I have? Different types of leukocytes. First of all, leukocytes in broad way, it has two types, granulocytes and agranulocytes. Granulocytes means which contains granule, A means absence, agranulocytes which doesn't have any um, granules or cytoplasmic, yeah, that's called the cytoplasmic granules. So granulocytes, again, it contains granules in the cytoplasm and it can be stained. The granules can be stained with the stain. They have possess lobed nuclei. And what are the granulocytes? There are three types of granulocytes, all end in phils. Whatever end in phils are the granulocytes, neutrophil, eosinophil, basophil. These are the granulocytes. They have the granules in there. Their nucleus is lobes, different lobes. And we have the agranulocytes. We have two types of agranulocytes, lymphocytes and monocytes. Lymphocytes are responsible for the body defense and monocytes also transfer to macrophage. When there is a site of infection, they have to go and do the function. So agranulocytes lack visible cytoplasmic granules. Nuclei are spherical, oval, or kidney shaped. That's fine. And it includes lymphocytes and monocytes. So how you are going to remember what is the highest number of WBCs to the lowest number of WBCs it can come. So it never let monkeys eat banana. Neutrophil is the highest one. The lymphocytes is the next one. Then after that is the monocytes. then the eosinophil and the basophil. So you have to remember this one. As per the list of abundance, how the WBCs are available. So remember this again. I am repeat. Never for neutrophils, let for lymphocytes, monkeys to monocytes, eat for eosinophils and basophils bananas. Never let monkeys eat banana. So these are the different types of cells. So we are going to learn how it originates. Erythrocytes, the basophils, you can see the granules here. Eosinophils, granules. Basophil has S-shaped nucleus. Eosinophil has bilobe nucleus. It's like 8. Then eosinophils has multilobe nucleus. Then monocytes also kind of bilobe nucleus, but it has no granules. And we have the lymphocytes. That's another type of blood cells. It doesn't have the granules. Monocyte doesn't have the glands. It's full of color. And we have the platelets and erythrocytes. So now neutrophils. Neutrophils, when you stain them, cytoplasm stains pink and contains fine granules. Deep purple nucleus contains 3 to 7 lobes. These neutrophils, you can see 3. It can be, lobes can be from 3 to 7. What is the function of neutrophils? Function at phagocytes at active sites of infection. We learned yesterday. So they go and they phagocytize the antigen. And number increase during infection. And it has 3,000 to 7,000 neutrophils in a cube millimeter of blood. If you guys can remember, that is going to be great. Otherwise, what you can say. And here is how much is the total population of the WBCs? 40 to 70%. Total WBC in the blood is only 1%, less than 1%. And in that 1% of WBC, majority is the neutrophils. That is the most abundant white blood white blood cells in the blood is 40 to 70%. Then eosinophils, they so their cytoplasm turns pink. This eosinophils is more red and it has more coarse cytoplasmic granules. I already told you, showed you it. Nucleus looks like 8. Shape is like 8. And nucleus turns blue red. Granules are red and their cytoplasm is blue red. So you can see is 8-shaped, large, coarse eosinophil granules, and background you can see the things is didn't came properly is a little bit blue in the color. What is the neutrophil? You have this granules and its cytoplasms also, but this is the very coarse granule. You can see this is the fine granule and this is the coarse granule. Neutrophils has lobe minimum 3 to it can be up to 8 lobe nucleus. What is the function of the eosinophils? Eosinophils is the only cells it kills the parasitic worm and play a role in the allergy. Also, it is active when there is an allergic reaction, but also it kills the parasitic worms. So only eosinophil can kill the parasitic worm. If there is a choice that whether the function of the eosinophil is to produce antibody to kill the parasitic worms and kill the and active in allergy attacks. What is going to be your answer?
Eosinophils.
Eosinophil, this is the eosinophil. But if I tell the eosinophil's function, number one is allergy attacks, number two is parasitic attack against parasitic worm, and number three is do from the blood clot. What is going to be the answer?
Number parasitic worm.
One,
Parasitic worms. Yeah.
allergy and
Yeah, it's going to be the first one is the parasitic worm.
Parasitic
If
worms.
there
Yeah.
is no option for the parasitic worm, but they give the option for the allergy and the other options, then you can took the allergy. Okay. 100 to 400 eosinophils in a cubic millimeter is nothing. It's 1 to 4% of the WBCs. And then we have the granulocytes basophil. Basophils purse out large blue-purple organelles. U or S-shaped nucleus stains dark blue. What it does, it releases histamines against the allergy attack. Okay, so histamine. So, their nucleus, their purple blue, yeah, their nucleus is U or S-shaped, and it releases histamine. This is a mainly allergy. This only the basophils fights against the allergy. So it releases histamines. It's called the vasodilator at the site of inflammation. What type of anticoagulant? So in the body, we have only one kind of anticoagulant. It's the heparin. Questions come sometimes. Do you have the anticoagulant? What type? Is the anticoagulant name is heparin. And we have 20 to 50% basophils in cubic millimeter of the blood. Lymphocytes, that are defense cells. Lymphocytes, cytoplasm is pale blue, dark purple, blue nucleus. Lymphocytes, dark purple nucleus and stains blue. And they are the agranulocytes. As we tell, monocytes and lymphocytes are agranulocytes. They doesn't have any granules. They function as a part of the immune response. We have two types of lymphocytes. We have discussed it several times. Beta lymphocytes, which produce antibodies, and T lymphocytes are involved in graft rejection. That we have learned yesterday. What type of grafts? So graft rejection, fighting tumors and viruses. When there is any problem like this, then these lymphocytes take action. So beta lymphocytes is mainly responsible for producing antibody, and T lymphocytes is act when there is a tissue transplant. So whether it's going to reject or accept, it depends upon the tissue T cell function, T lymphocytes function. So that's why I said the T lymphocytes are involved in graft rejection, fighting tumors and viruses. The lymphocyte amount in the blood is 1.5 to 3,000 lymphocytes in a cubic millimeter of blood. So only 20 to 45% of blood WBCs. So another next continued type of agranulocyte is monocytes. It is the largest white blood cells. So okay, what is the abundant or maximum white blood cells in the blood?
Neutrophils.
Good. And what is the largest WBC? Is the monocytes. So it's the largest white blood cells. It has gray-blue cytoplasm. It has dark blue-purple nucleus in often kidney-shaped. And as we learn that it functions as a macrophage. And it helps in the fighting chronic infection, long eosinophil. Whenever there is allergic reaction or parasitic reaction, they will come and do the job and they will stop. But lymphocytes, they are active when there is a body defense and other things. But you have chronic infection, no cell, they just forgot that time the body. What is that? Chronic. Only this agranulocytes, monocytes take action for the chronic infection. Now, next one is platelet, is the blood clotting cell, help in blood clotting. Derived from ruptured multinucleate cells is called the megakaryocytes. So from where platelets develop is developed from the megakaryocytes. You have to remember the name, megakaryocytes. Platelets derive from that. What is it is needed for clotting process, and platelet count ranges from 150,000 to 400,000 per cubic millimeter per cubic millimeter per blood. So if that is high or low count, so then probably if it's a low count is a problem, generally people come for the transfusion. That's time. Oh, it's not showing it. So how where we have learned from where the blood cell forms in the red bone marrow, right? All blood cells are derived from a common stem cell. Stem cells which stay in the bone marrow is called the stem cells. The main stem, like a stem of a plant, main stem is called the hemocytoblast. Blast means it's going to blast, generate. Cyto means cells and hemo means blood. So hemocytoblast is in one name. So it is all blood cells are derived from a common stem cell is called the hemocytoblast. Hemocytoblast differentiation, it differentiate to form different types of cells. Lymphoid stem cell produce lymphocytes and myeloid, and then hemocytoblast has two different sectors. In one side, in one pathway, it's produce the lymphocytes, and another pathway use the called the myeloid stem cells to produce all other formed elements. So see here, what is that? So you see, hemocytoblast, that is the stem cells, precursor stem cells in the bone marrow, red bone marrow. From here, all the different types of WBCs are produced. So it has two direction. One is lymphoid cells and another is myeloid stem cells. Okay, the lymphocyte cells is only going to produce lymphocytes, but then from the myeloid stem cells, it produces so many different types of white blood cells. What did this one? What did this one? What is the name of blood cell formation? Is called hematopoiesis. Blood cell formation, all type of blood cell formation is called hematopoiesis. Formation of red blood cells is erythropoiesis. Formation of WBCs is called leukopoiesis. Formation of platelet is called the thrombopoiesis. So you have to remember this terminology. Hematopoiesis versus erythropoiesis, leukopoiesis, thrombopoiesis. Hematopoiesis is the blood cell formation. Any type of blood cell formation occurs in the red bone marrow and red bone marrow inside the brain. All blood cells are derived from, we are already told, from the common stem cells. It's called the hemocytoblast. Then hemocytoblast differentiate. It differentiate in the lymphoid, what I show in the diagram, and the myeloid. Lymphoids, from the lymphoid stem cells only produce lymphocytes, and myeloid stem cell produce all others. Erythrocytes and all other 4 WBCs and platelets. So again here, myeloid stem cells, lymphoid, myeloid. From the myeloid, it produces erythrocytes, basophils, eosinophils, neutrophils, then monocytes, platelets, and erythrocytes. Formation of erythrocytes. They are unable to divide, grow, or synthesize proteins. They cannot do anything. They doesn't have the nucleus, right? So they cannot divide, they cannot grow. They are only loaded with hemoglobin and acts as the delivery person of the oxygen and carbon dioxide. So that's why it's wear out in 100 to 120 days. This is another important next one. When RBC got destroyed, so they worn out into the pieces, right? Who destroy them or who take care of the cleaning of the destroyed or worn out RBCs? That is the liver. Oh, sorry, spleen or the liver. The worn out the RBCs goes to the liver and spleen, and then the liver or spleen cells worn out they take care of the worn out RBCs by the phagocytosis. So these questions come after the worn out WBCs are eliminated by which part? Spleen or liver? And then lost cells are replaced by the division of hematocytoblasts in the red bone marrow. So WBC is always, RBC is always getting, after 120 days is getting worn out as they doesn't have the nucleus, and new RBCs is always producing. So it's a cycle. Every day, some are earning out or getting breakdown, and new are produced because you have to make the balance. So the production or the formation of the erythrocytes, that's the RBCs, is regulated or controlled by a hormone is called the erythropoietin. But this erythropoietin hormone is not produced in the bone marrow where the erythrocytes are formed. Where it produced? It's produced in the kidney. So erythropoietin is a hormone. So we learn the endocrine gland. Kidney is one kind of kidney. So what it does, it produces the hormone, and the hormones is transported to the blood to the target organ. So for the formation of erythrocytes or RBCs, we need the erythropoietin hormone. The erythropoietin hormone is produced in the kidney, then transferred through the blood, and ultimately it goes to the bone marrow, where the production or synthesis of the RBCs takes place. So when there is a low, when kidney produce, how kidney sense that I have to produce the erythropoietin? When the RBCs in a regular way worn out or destroyed, there is a low oxygen, because low number of RBC means low number of oxygen, low hemoglobin, low oxygen. So that is called the hypoxia. When there is a low or reduced oxygen level in the blood, Kidney understand, oh, okay, so I have to synthesize erythropoietin and secrete it so that body needs more erythrocytes. And if the erythrocytes I can produce the erythrocytes, then there will be more oxygen, enough oxygen in the body. So what is called the homeostasis is the maintained by negative feedback from the blood oxygen levels. Why is called the negative? Because when the amount or the concentration of the oxygen or the level of oxygen is less, then kidney is producing the erythropoietin. That is called the negative feedback. When is we are going to learn later positive feedback, when some, say for example, erythropoietin level is become too high, but the blood doesn't need that much exercise. This is called the, again, it shuts down. So when the concentration of something is high and it stops the production or synthesis of the function, that is called the positive feedback. And when the level is low and is regulating or producing something, is called the negative feedback. So here, formation of the erythrocytes or RBCs is governed by the negative feedback mechanism, by the production of erythropoietin hormone from the kidney when there is a low oxygen level. Clear?
Clear, thank you.
Okay. Okay, so you got it, right? So these are the pictures how it's showing whatever we told you. Stimulus, low blood oxygen carrying ability, then kidney produce the erythropoietin. Erythropoietin goes travel to the bone marrow, red bone marrow, then it enhance the erythropoiesis, produce the erythrocytes. These are the stepwise they are showing it. We doesn't need to, because you guys know whatever. Now we are going to learn about the white blood cells. White blood cells is also white blood cells and the platelets. These are also controlled by hormones, everything. In general, hormone is a very important regulator for all body functions. But there are not only one hormone, there are several hormones. So what is called colony stimulating factors is called the CSFS, and interleukins prompt bone marrow to generate leukocytes. It's not like the erythrocytes production, but when there is a need. So when there is a low WBC for anything, or there is a huge for the infection, there is a huge number of WBC. So the regulation, the homeostasis balance is by production or stop production of the colonistable factors. It's called the CSFS and interleukins. This prompts bone marrow to generate leukocytes. And what is the another, like the erythropoietin? Platelets, the name is thrombocytes. Another name of platelets is thrombocytes. So which hormone regulates or stimulates the production of platelet is called the thrombopoietin. So I told you at the beginning there is a difference between homeostasis. Majority of the students make mistakes. Right note. Homeostasis is the balance, internal balance. And hemostasis is the stoppage of bleeding. Look at the question, is that homeostasis or hemostasis? Then look at the answer and give the answer. Don't put the answer of the hemostasis to the homeostasis and answer for homeostasis to hemostasis. That mistakes happens lots from the students, because I understand during the exam you have run against the time. Sometimes you overlook. That is, you have to be very careful and attention detail. That's one of the major important factor in our job. Okay, so hemostasis is a stoppage or bleeding resulting from a blood break of a blood vessels. It has three phases. One is vascular spasm, then platelet plaque formation and coagulation. So how it happens? Vascular spasms is vasoconstriction, causes blood vessels to spasm. What happened? It start constriction means contract, it constrict. And when they constrict, this spasm which constrict the blood vessels means it's narrowing the blood vessels to decrease the blood loss. That means it's narrowing the spasm is constriction and it's narrowing the blood vessels so that we can decrease the blood loss. And then is the C. How it happens? Step 1, vascular spasms occur. This is the big one when the smooth muscle contracts. Smooth muscles, inside that we have the tunica interna and in between you have the tunica media. That is the main muscle cells when we do the anatomy of the vessels. So what happened? These constrict or contract, and when it contracts, it narrows the opening of the blood vessels, causing less blood flow. Then is the platelet blood formation. So what is that? Collagen fiber are exposed by a break in a blood vessel. So when there is a break, these collagen fibers are exposed. So then collagen fibers, then platelet becomes sticky and cling to fibers. So collagen fibers comes there and platelet from the blood, it become stick to the collagen fiber. And when they become stick in that area, they start releasing some chemicals to attract more platelets. Okay, I am giving you the signal. What is the signal? It gives release some chemical. And with that chemical is giving the signal to platelets. Okay, guys, there is a rupture in the blood vessel. Please come. We have to take care of that. We have to make a plaque formation so that we can stop the bleeding. So then platelets piled up one up another to form a platelet plaque. See. Platelet is these are the injury vessels. These are the white one is the collagen fiber. So first it constrict, then platelet plaque formation. Collagen fiber, on the collagen fiber, these are the platelets are coming and sitting. The small platelets coming and sitting one over another, like you are putting one another over another, and it's making a huge plaque. And with these plaques start the basis of the coagulation. Then what happened? These injured tissues release tissue factor, TF factor. You doesn't have to remember all the factors name, but you can set coagulation. Injured tissues release tissue factors. There are many factors, but the main one you have to remember, prothrombin. This is important. Prothrombin activator converts prothrombin to thrombin, an enzyme. So here now the platelet gathers here inside the platelet, what is called, it converts the prothrombin to thrombin, the enzyme. And then what the thrombin does, we have fibrinogen proteins in the plasma. So what happened? The thrombin joins fibrinogen proteins into hair-like molecules in the insoluble fibrin, like a mesh work. And this is called the basis of clot formation. Now see this one. So how it happens. So coagulation events occur. There collagen, then platelet plaque formation. Then it has been in the injured tissue already from the prothrombin. It converts to an enzyme called thrombin, and this thrombin is now bringing the fibrin. This is the threads, fibrin threads, one over another to make a mesh-like structure. That is the clot, and it stays until the total bleeding procedure is done. So how it looks like, the clots. This is the RBCs is trapped in this clot. So what is the blood clotting? Normal blood clotting time, 3 to 6 minutes. the clot stays there. When you have some clot, you can see some black things scarred, like, so that is called the clot. The clot remains there until the new tissue has been generated, because that's the cut, right? There is a hole. So if the clot doesn't stay there, there will be leakage of the plasma. So clot remains as endothelium degenerates, and clot is broken down after tissue repair. When the all tissue is repaired, there is no more leakage, the blood vessel become intact, then the clot is broken down after tissue repair. So now couple of terminology related to clotting. What is thrombus? A clot is an unbroken blood vessels, can be deadly in areas like the heart. So thrombus, if any clot happens near the heart, it can be deadly, because it will totally stop the, or it will hamper the blood flow or stop the blood flow. That is when you have blood clot in the brain, is going to cause you stroke. So heart in the blood clot in the heart is can cause you heart attack, and it can stop the heart. What is the embolus? This is very difficult. Is thrombolus that breaks up and floats, is from the clot. Sometimes it breaks away. It's a floating clot. It can move in the bloodstream, and it can cause anywhere. It can clog vessels, critical areas such as brain. That's stroke. So embolus, if it's a big one or small one, that is the floating or moving blood clot, is called the embolus. What are the different bleeding disorders? Thrombocytopenia. When cytopenia, penia means always less, right? When there is a platelet deficiency, then it takes long time for the blood to clot, because there is not sufficient platelet which can do the job. Even normal movements can cause bleeding from small blood vessels that require platelet for clotting. Hemophilia, hereditary bleeding disorder. When it's a genetic, it's called the hemophilia, because they miss the genetically, they miss the normal clotting factors. Now coming to blood group and transfusion. Large losses of blood. When you need the transfusion, either you are too much anemic, or huge loss of blood have serious consequences. Loss of 50 to 30% causes weakness. Loss of over 30% causes shock, which can be fatal. You can be in the coma, you can be heart attack, you can be, you lose your, sorry, not heart attack, you can be lose your vital signs. So transfusion are the only way to replace the blood quickly. But transfused blood must be of the same blood group. What are the blood groups? Blood contains genetically determined protein. Inside the blood, we have different proteins. It's called the blood group proteins. What are that? It's the antigen, a substance the body recognizes as a foreign, may attack the immune system in the blood. So it's not compatible one. The blood is going to be agglutinate or clumped. So antibodies are the recognizer. Blood is typed by using antibodies that will cause blood with certain proteins to clump, is called the agglutination. So when you are going to mix with specific group of blood with their antibody, is going to clump or agglutinate. That way you can understand which blood group the person is or the patient is. So there are over 30 common blood cells antigens. That is, we do when we need the typing, blood typing. But most vigorous transfusion reactions are caused by ABO and RH blood group antigens. The majority, as per the blood group, immediately we do the ABO grouping and RH. That is the main important blood group system. So how ABO blood group is be determined? Depending upon the two types of antigens, A and B. That is, O means there is no antigen. That is, the lack of this antigen is called O. O doesn't have any antigen. So we have type A, type B, and type O. O when there is nothing. Okay. When we have both antigens, we call the AB. When there is only presence of antigen A, is called A. When there is antigen B, is called B. And when there is nothing, lack of antigens A and B, is called type O. So we have four major ABO blood type. Then we have the RH. So, okay. So now who can receive which blood? Blood AB type is called the universal recipient, means they can receive the blood. AB blood grouping is the universal recipient because it has A and B, so it can get the blood from A, it can get the blood from B, it can get the blood from the AB, and it can get the blood from the O. So they are the universal recipient. Blood B can get the blood from the B and O because O has no antigen. So it's a universal donor. Anyone by closing their eyes, if there is no specific their blood group matching, they can get the blood from O. So that's why it's called the O is the universal donor. And you can see probably when you are doing the blood grouping testing, we have majority of them is O. Rare is AB. Okay. So we can get only the blood from B and O group. A can receive blood from A and group. The O is poor, very, they can only receive O blood group because they have nothing, no antigen. So they only depend on the O blood group. So that's why it's called the universal donor. They can give to everyone O, but they can only take the blood from the O blood group. So see how blood grouping AB. What type of antigen it has? A and B. They doesn't have any antibodies here. So universal recipient AB, AB and O. B blood group has RBC. The antigen is B, but in the blood is the antibody A. So that's why it doesn't. If it has anti-B here, it's going to clump. So anti-A cannot interact with the B. So when there's a B, means it has the anti-A. And when there are both antigens, there is no antibodies, plasma. Okay, this is on the RBC. Antigens are present in the surface of the RBC and antibodies are present in the plasma. So they can receive the blood, the B group can receive the blood from the B and O. Same A, they have the RBC antigen A, and in the plasma, they have the anti-B, and they can receive the blood only from the same group A and O. And O, they doesn't have any antigen. So they have both antibodies, anti-A and B. So immediately, if you give the A blood group or B blood group, it's going to start clotting, agglutination or clumping. So that's why it's called the universal donor. So what is the RH blood group? This is mainly further related to the pregnancy, named because presence or absence of one of the 8 RH antigen. This is called the RH antigen. Is called the agglutinogen D. RH. That's why it's called the D. We call the RH as D, ABO and D. So that was why it's called the RH because it has been first defined in the rhesus monkey. So that's why it's named as the RH blood group. Most Americans are RH positive. That means they have the RH group. What happened? problems can occur in mixing Rh positive bloods into a body with Rh negative blood. So that's why you need the ABO blood grouping. If somebody's AB blood group, AB positive means Rh positive, they can get blood from the AB positive, A positive, B positive, or O positive. They cannot get the blood from the A negative or B negative or AB negative. Then it's going to start clumping. Same thing for each one. So mainly it happens, danger occurs only when the mother is Rh negative and dad is Rh positive. And children's inherits the Rh positive factor. Because one is positive, one is negative. So it started interacting. So that's why when there is a mom is negative and dad is positive, for the pregnancy, they got miscarriage. So that's why you have, now it has been developed, not long before, not now, ROGAM, is that the short, which can prevent the buildup of anti-Rh antibodies in mother's blood, so that even there is Rh positive, this ROGAM is preventing the formation of antibodies. That means this ROGAM helps to prevent the interaction between mom's Rh negative with the dad's Rh positive factor. So Rh dangers during pregnancy. The mismatch of an Rh minus mother carrying an Rh baby. Mother's own blood is Rh negative, but mom's is carrying a baby which is Rh positive because one came from dad and one came from mom. But Rh positive is the dominant one. So baby is Rh positive, and which can cause problems for the unborn child. First pregnancy generally doesn't have any problem because the baby is there. Why? There is an exchange in the placenta. So first pregnancy, not at the pregnancy, when they're delivering the baby. So first pregnancy generally proceed without problems, no problems. But
I have a
I know,
question, Miss.
yes,
So in this case with the RH, is it better than that you know the blood type of the person before conception?
yes,
Because
100%.
yeah,
Not only that,
yeah,
blood grouping also important. Yeah.
yeah.
Blood grouping is okay, but for the baby, Rh factor is very, very important, whether you are positive or negative. Yes, yes.
Okay,
If you know,
thanks.
even if you know, it's okay. Rh positive or negative, even you know, there is a protection. You have to tell the doctor that my husband or my boyfriend or whoever else, the man is Rh positive and I am Rh negative.
Mhm.
So then they can give you the Rogan shot. They will test your
Okay,
blood grouping and they can give you the shot. Okay, so
good.
that's why the first, the first
Yeah.
pregnancy, because there is no connection between the blood, right? So first pregnancy is
Mhm.
okay, but immune system is sensitized after the first pregnancy, because in the second pregnancy, what happened? Mother immune system now produce antibody because mother's blood is exposed when it's delivering the blood everywhere, right? And the baby is coming out. So mom's blood has been exposed to the Rh positive bloods from the baby. So now inside the mom, it has the Rh positive and it's always produce the antibodies to attack the Rh positive bloods of the newborn. It's called the hemolytic disease of newborn or HDN. So what is called the blood typing or blood grouping? We take the blood samples and mix with different antibodies, anti-A and anti-B serum and also the anti-D. Coagulation or no coagulation leads to the determination of the blood type. It's clotted or not clotted. So typing for ABO and Rh factors is done in the same manner. It's called the cross matching. What is cross matching? Testing of agglutination of donors RBCs by the recipient serum. So cross matching means when you are testing for the transfusion, right? Transfusion. What? You are taking the Donor's RBC, donor's blood, and who is giving the blood, we are taking the serum from them and testing it to make sure is there agglutination or not. So how it agglutinate? So this is serum. This is the blood cell. This is the blood cells. Okay. So it's type AB, contains antigens A and B. So it has in the surface is that antigen A and antigen B. When we are putting the anti-A is antibody against the anti-A and anti-B, it started the clumping. See, you can clumping. So that means when it clumps, it belongs to that blood group. Okay, so in the RBC, we are taking the blood, we are putting in the slide, and then we are putting the anti-A and anti-B. Anti-A and anti-B in general inside the blood, it has in depending upon the what blood group you are. So type B is the blood cell erythrocytes. When we are putting the anti-A, it's doing nothing. But when you are putting the anti-B, it's starting agglutination. That means it's type B blood group. Same thing like A, and in O, no agglutination because there is no antigen. So even what you give any antibody is not going to react. So when we did the, somehow do we discuss the in the bone marrow when you are doing this one. So what the mainly fetal blood cells form in the liver and spleen, because at the beginning of the fetal they doesn't form the bone that time, right? So there is no road bone marrow or anything. So fetal liver and spleen are the early sites of blood cell formation. Then slowly as the bones develop, bone marrow takes over the hematopoiesis. The 7 month, around the baby 7 month, the bones has been formed properly and bone marrow is developed. So from the 7 months, bone marrow slowly take over the charge for production of the blood cells. Fetal hemoglobin differs from the hemoglobin produced after birth. What is physiologic jaundice result in the infants in which liver cannot read the body of hemoglobin breakdown products fast enough. So that's the physiological. They cannot get rid of this one. Okay. We done the blood. Any questions?
Break time.
Break time.
I know break time. I understand. You had an question. I know the. I'm sorry because if I just do in the break because we have to go to the more. So it's just a lot of information here. Okay.
Yes.
So yeah. So go for break.
Okay.
Thank you.
Thank you, Miss.
Welcome.
20 minutes.
Yes.
Thank you.
Okay.
Thank you.
Welcome.
Ma'am,
Who?
ma'am,
Who is that? Who is talking? Rugiato. Yes. Tell me. I can't hear you. I can't hear you, dear. Rugiato, I cannot hear you. I can see you, but I cannot hear you. Can you write it down? Yeah, write down. Message me. Message me. Message. Then I can understand. I cannot hear you. Others can hear me?
Yes, we can hear you.
Rugiato, send me the text message.
can you hear me?
Who is that? I cannot hear you, Giato.
Can you hear me now?
Yes, now I can hear you. Yeah, yeah. What's your question?
Okay.
Again, no answer, no voice. I cannot hear. I don't know. I can see your lip is moving, but I don't understand what you are telling. I cannot hear. Some problem in your microphone.
Miss?
I hope everybody is back. Now let us go quickly the video and then we are going to move to the next one.
Yes.
Yes.
Can I ask a question about the about the RH, because it's very interesting. I just have one question.
Yes.
If, say for instance, you are like A or AB positive, and do you have the RH in you, or is it just RH?
I didn't get your point.
Okay,
What you
like
are
say,
asking?
okay, is the R, because I was reading on the internet just now, it said RH positive and
That
RH negative can be a
negative
danger,
means you
right?
does, yeah, RH negative. That if you have in the body, that is mainly mix up between the during the pregnancy time. Otherwise, in either in your body is going to be RH positive or RH negative. Only
Anybody
when a
can
baby,
have.
yeah, anyone, you may be RH positive or negative. Anyone, either positive or negative.
Okay,
You
that's what
cannot
I
be both.
figured.
Mhm.
Yeah, that's what I figured.
Only the mixing of blood during the pregnancy, during the delivery, that happens.
Got
From the
it.
RH, yeah.
Yeah, thank you.
Okay.
Thanks for clarifying that.
You're welcome. So let us start the video and then we'll move on to the next one. I think the immune system. Okay. I think everybody is back now. I'm starting the video.
Wix is your platform to perform on.
Don't take this the wrong way, but you're pretty replaceable. When it comes to your body, science has figured out how to hack, synthesize, or replace a surprising amount of its parts and processes. We have implants to keep heartbeats steady and steel rods to mimic bones. We've got drugs that can replace hormones and antibiotics to cover for your immune system. And pretty soon, you'll be able to just 3D print a new ear if you need one. Really. The one thing we absolutely cannot manufacture, despite what True Blood would have you believe, is blood. And yet blood is a thing that we all need. And sometimes, because of injury or illness, we need extra blood. In fact, every 2 seconds, someone in the US needs a blood transfusion. This could be a victim of a car accident, someone undergoing surgery, or a cancer patient who needs new blood to maintain their health during chemotherapy. And because we can't grow it on trees or make it in a lab or even store it for all that long, the blood that people need, nearly 16 million pints a year in the US, has to come from people who have donated it. So, let's talk blood, shall we? The meal of choice for vampires and female mosquitoes, blood is red, sticky, salty, and kind of metallic tasting. It is indeed thicker than water and super viscous, which is why Hitchcock used chocolate syrup as a stand-in a certain classic shower scene. For most purposes, blood comes in 8 different types, and it accounts for about 8% of your body weight. You might remember from our episodes on tissues that blood is a type of connective tissue, which means it's made of living cells suspended in a non-living matrix, which in this case is the fluid ground substance called plasma. And of course, one of blood's main missions is to transport and distribute oxygen and nutrients and waste products and hormones around the body. But it also helps maintain and regulate body temperature, pH levels, and the volume of fluids in your body. Plus, it protects you from infection and from the loss of blood itself. Perhaps second only to your brain, your blood is the one component of your body that we haven't figured out how to reproduce, synthesize, or imitate. It's a part of you that is literally irreplaceable. It's Saturday and you feel like doing a good deed, so you head over to your local Red Cross for a blood drive. You get your finger pricked and then somebody directs you toward a lounge chair, swabs your inner elbow with alcohol, and then comes at you with a hollow needle. Once the bag is full, they usually take about a pint, you get unhooked and grab a cookie and a juice to replace the blood sugar you lost, and the whole process takes around 20 minutes. But for your blood, the day is just beginning. Soon it will be taken to a lab where it'll be tested for infectious diseases and separated into different parts before heading out to hospitals. So hold up. What exactly do I mean by different parts? Well, the blood that flows from your arm into that bag is whole blood, a mixture of cells and cell fragments called formed elements along with water and lots of dissolved molecules. A patient who needs a transfusion may only need some of those things and not others, so the parts are separated. Once your blood makes it to a lab, technicians put it in a centrifuge which spins it around fast enough to send the heavier components to the bottom of the tubes and bring the less dense elements to the top. In the centrifuge, 3 distinct layers emerge. Down at the bottom, you got a heavy red layer of erythrocytes, or red blood cells that carry oxygen and carbon dioxide. They make up about 45% of your total blood volume. Then you got this thin little whitish layer in the middle. Those are your warriors, the leukocytes, or white blood cells that defend your body from toxins and foreign microbes. And there are also the cell fragments called platelets, which help with blood clotting and make up less than 1% of your blood. Finally, up at the top, you see the yellowish plasma, which accounts for about 55% of your blood volume. Plasma is actually 90% water, but the other 10% is chock full of 100 different solutes, including proteins, electrolytes, gases, hormones, and waste products. The most abundant of these solutes are electrolytes, which you may have heard of as the secret ingredient in sports drinks, but they're really just positively charged cations like calcium, sodium, and potassium, and negatively charged anions like phosphate, sulfate, and bicarbonate. Together, these ions help regulate your blood's chemistry, maintaining its pH levels and proper osmotic pressure, and allowing other tissues to do their jobs, like making muscles contract and sending action potentials. But when measured by weight, the bulk of the solutes in your blood are really the plasma proteins. Most of these proteins, like albumin and alpha and beta globulins, are made by the liver and do things like balance the osmotic pressure between the blood and surrounding tissues and transport lipids and ions. Others run defense for you, like the gamma globulin antibodies that are released by plasma cells during an immune response, or fibrinogen proteins, which are vital to forming blood clots and stopping bleeding. All right, bleeding. I want to talk about that. Because for the very reason that I mentioned at the beginning, that we can't replace your blood with some synthetic wonder fluid, the last thing that your circulatory system wants is for you to fritter away your blood in some sidewalk scrape or kitchen accident. So it has a whole system in place to prevent you from losing too much of it through a process known as hemostasis. So imagine you're slicing up a nice garlic cheese bagel one morning and you lacerate the distal phalanx of your pollex. In other words, you cut the tip of your thumb. And now you're bleeding all over your breakfast. At the very first sign of rupture, the blood vessel actually constricts itself to slow the flow of blood through it. Then little cell fragments called platelets gather at the site of the injury, creating a plug that dams the breach and keeps the blood from leaking further. Now, these free-floating platelets don't clump together during regular circulation. That would be terrible. But when the endothelial cells lining a blood vessel wall tear, the underlying collagen fibers are suddenly exposed, and they chemically react with the platelets, turning them all sticky and glue-like at the scene of the injury. But that platelet plug still isn't as strong as it could be. It needs reinforcement to complete the clotting process. This reinforcement comes in the form of fibrin threads, protein strands that join together to make a sort of mesh that traps the platelets and blood cells. Eventually, the threads actually pull the opposite sides of the wound together to close the vessel wall so the endothelial cells can be replaced. Over a few days, the blood vessel heals and the blood clot dissolves, or at least that's how it's supposed to happen. People who suffer from disorders related to hemostasis may have trouble with unwanted clotting or the inability to form clots. In the family of disorders known as hemophilia, a patient can usually complete the first 2 steps of hemostasis just fine, but they can't make an effective fibrin clot. So it's not that they bleed more than anyone else, it's just that they bleed longer, which I guess means that they bleed more. As a result, they may need frequent blood transfusions throughout their lifetime, which brings me right back around to that Saturday morning blood drive. Another thing you're going to need to know before you give blood is what type you have. Do you have A, B, AB, or O? These different types all do the job equally well, they just sort of have a different flavor related to your immune system. All the cells in your body have a plasma membrane with specialized glycoprotein markers on them that act like name tags or labels, sort of like, this cell is property of Hank. These markers are your antigens, and your body's immune system is totally fine with your particular antigens, but if it detects antigens from someone else's cells, including viruses or bacteria, then it'll send out antibodies to bind to those markers, often to tag them for destruction by the immune system. Your red blood cells have specialized antigens on them called agglutinogens that activate antibodies that work by binding invading cells to each other, which causes coagulation, or the clumping of blood. Which agglutinogens you have on your erythrocytes defines your blood type, but they're classified in 2 different ways. In the most important blood classification, the kind people are most familiar with, there are only 2 kinds of agglutinogens, simply A and B, and your blood can either have one or both or neither of these molecules. So the name of your blood type refers to what kind of agglutinogens you have or don't have. A type has A A antigens, B type has B, AB has both, and O has neither. So why do you need to know what type you are before you give or receive blood? Well, like I mentioned, if you have either of these antigens, your body will be fine with it because it doesn't produce any antibodies that label it for attack. So if you don't have a particular antigen on your blood cells, say the type B, then you do have antibodies that are going to label those B antigens for attack should they enter your space. So AB type folks are called universal recipients because they have both antigens and therefore no antibodies for either. So they can accept A or B or AB or O blood. Meanwhile, O type doesn't have A or B antigens, so those folks have antibodies for both. That means that they can only accept other O blood. And yet that lack of antigens means that type O blood can mix with other types of blood without getting attacked, which is why it's known as the universal donor. But just to complicate things a little bit more, you've got a whole other set of antigens with totally different protocol. These are your rhesus or Rh antigens, named after the species of monkey they were first identified in. Much like A and B, you either have the Rh antigens, in which case you're Rh positive, or you don't and are Rh negative. Most of the population is Rh positive, so they don't have the anti-Rh antibodies, which means they can accept either positive or negative blood, but negative types should stick to just the Rh negative blood. And since the presence of AB antigens is controlled by different genes than the Rh ones, we end up with 8 different blood types, 4 separate groups, each with 2 variations. And now, hopefully, you understand why it's so hard to replace blood, and why true blood is not true. I've not actually ever seen that show. Along the way, you also learn the basic components of blood, including erythrocytes, leukocytes, platelets, and plasma, as well as the basic process of hemostasis that stops bleeding, and how antigens are responsible for the blood type that you have. Thanks to all of our Patreon patrons who make Crash Course possible through their monthly contributions. If you like Crash Course and want to help us keep making it for free for everyone,
Save money and time with Sign Now, a powerful e-signature solution. Digitalize and simplify any
I feel like I haven't spent nearly enough time lately talking to you about all the stupid and dangerous things that you can do to your own body. So let's talk about doping. You probably have heard of this thanks to Lance Armstrong, who secretly messed with his own blood so that he could illicitly win the Tour de France 7 times in a row. You might be dimly familiar with the fact that doping isn't like shooting steroids, but it is still cheating, even though, like, why is it cheating and how does it work? And is it even possible to make your blood better at being blood? In other words, how can some people treat or mistreat their own blood like it's some sort of drug? Short answer, because your blood is incredibly powerful stuff. And its power rests largely in your erythrocytes, or red blood cells. They're the most abundant type of cell in your blood, accounting for nearly 45% of its volume. Every time you take a breath, they pick up oxygen in your lungs and distribute it through your body and then grab carbon dioxide and bring it back to the lungs where it can be exhaled. The main mission of erythrocytes is to keep your body fed with oxygen so your muscles can do their thing and your brain can continue to think and feel and boss around your various parts. But you don't want to mess around with your red blood cells because erythrocytes are weird characters. They go places that other cells won't. They purge themselves of their most precious inner belongings, preferring instead to live as hollow shells. Because of the crushing demands of their job, they don't live very long, and just like with your blood pressure, too much of these good things can turn bad quickly. So the erythrocyte must be respected. It is not for doping or for dopes. Despite their prominent role in some international sports scandals, your red blood cells are fairly simple and unassuming little cells. They've got a distinct biconcave shape, which just means that they're concave on both sides, making them look kind of like a breath mint, a tiny, bloody breath mint. And while they do have a plasma membrane, they don't have a nucleus and don't have most of the parts that other cells do. So they're basically just glorified protein-filled phospholipid bilayer sacs. But there's still another great example of that harmony between form and function. For one thing, that biconcave shape gives them a large surface area that's ideal for gas exchange. It also makes them flexible and able to change shape as they squeeze through tiny capillaries with diameters smaller than the cell itself. Of course, all that squeezing and twisting is hard on a cell's membrane, and that, combined with their general lack of organelles to help repair the membrane, means these cells don't live very long, surviving on average only 120 days. But they sure work hard while they're alive. And their work is mostly in gathering and transporting oxygen. They're able to do this because if you don't count their water content, red blood cells are 97% hemoglobin, a molecule that easily binds to and releases oxygen. It's like an oxygen sponge. Every hemoglobin molecule is really made of 8 different component molecules. 4 are a red pigment called heme, and 4 are a protein called, you guessed it, globin. Each globin is a globular polypeptide chain, hence its name, and proteins, you'll probably remember, like to bind to stuff. So each globin has its own personal ring-shaped heme molecule, and in the center of that heme is an iron atom, kind of like a cherry on top of a protein and pigment sundae. It's that iron in the center of the heme that makes our blood red. Incidentally, not all animals have red blood, because not all animals use hemoglobin to move oxygen. For example, most mollusks like squids and snails have blue blood because it contains hemocyanin, a copper-rich protein pigment that turns blue when exposed to oxygen. But iron is what we're stuck with, and I have to say it's great at its job because each iron can bind with one whole oxygen molecule. And that oxygen really adds up. Since you have 4 iron atoms in every molecule of hemoglobin, and every red blood cell contains something like 250 million hemoglobin molecules, that means that each one of your tiny floppy red breath mints can grab about 1 billion molecules of oxygen. Exactly how they transfer oxygen and carbon dioxide from your tissue cells is something that we'll get into more when we talk about the respiratory system. But if you're wondering why all this hemoglobin can't simply skip the red blood cell rigmarole and just run around naked in your blood, it's because free-range hemoglobin would actually thicken the blood, making it so viscous that it would impede blood flow. This also happens to play a part in blood doping, which, stick with me, I will explain in a bit. So what does the brief but glorious 4-month life of an erythrocyte look like? Well, remember when I said that a red blood cell doesn't have a nucleus? And maybe you thought, hey, wait a second, how can it even be a cell without a nucleus, or DNA? First of all, nice catch. But actually, erythrocytes do start off with a nucleus and DNA. They just get rid of them because their entire purpose for existing is to schlep around hemoglobin and oxygen, and they want the extra room. The whole process of forming red blood cells, called hematopoiesis, happens in your red bone marrow, which is mostly made of reticular connective tissue that's snuggled up to special capillaries called blood sinusoids. In short, the process begins with a hemocytoblast, or a specialized stem cell, which soon differentiates into an early erythroblast. Then it starts making a whole bunch of ribosomes, the organelles that manufacture proteins. And in this case, the ribosomes start cooking up tons of hemoglobin as the cell transforms into a late-stage erythroblast. When it's got enough hemoglobin, it suddenly jettisons most of its organelles, which causes the cell walls to collapse a little, giving it its biconcave bloody breath mint shape. Now you're left with a reticulocyte, which is pretty much just an early erythrocyte that still has a little group of ribosomes left called a reticulum. So far, this whole journey has taken about 15 days. When the reticulocyte is finally just about bursting with hemoglobin, then it leaves the marrow and enters the bloodstream, and a couple of days later, when the last of the ribosomes have degraded, you've officially got yourself a mature red blood cell. And that cell travels around your body doing its job for a few months before it gets old or damaged and needs to be replaced. Now, maintaining the balance between production and destruction of these cells is crucial. Too many will make the blood too viscous and difficult to pump, and too few leads to oxygen deprivation, or hypoxia. The process of maintaining the right levels of red blood cells is regulated by a special hormone called erythropoietin, or EPO. It's produced mostly in the kidneys, but also in the liver, and is constantly circulating in the blood. If you're anemic, or hiking at high altitude, or hemorrhaging blood, or experiencing anything else that creates a drop in your blood oxygen levels, certain cells in your kidney will take notice and take action. And they can do that because they traffic in a signaling molecule called hypoxia-inducible factor, which monitors your blood's levels of oxygen. The way that this works is pretty cool. These special kidney cells need oxygen in order to break down that signaling molecule. So if oxygen levels in the blood are low, they can't turn the signal off. This means that the signal keeps going, which triggers the release of more and more EPO, which stimulates your red bone marrow to pump out more red blood cells to carry around more oxygen. As oxygen levels in your blood increase, the signal is degraded and EPO production slows. And EPO is a key player in blood doping too, but we're gonna have to wait a minute before we get there because I first want to get back to the fate of your hardworking erythrocytes. So if you're generating around 2 million new blood cells every second, you also have to dispose of about the same number of dead ones to maintain the balance, right? When these cells get old, they turn rigid and their hemoglobin start to fall apart. As they get stiffer, they can end up getting stuck in capillaries in your brain or heart, which would not be good. Luckily, you have certain channels to corral these dying cells, especially around the spleen, which some anatomists call
the red blood cell graveyard.
So these tired old cells get trapped and then basically ambushed by big macrophage white blood cells in the spleen, liver, and bone marrow, which break them down and recycle their various components. The globin proteins are broken down into their basic components, amino acids, which go back into the blood to be used by other cells for making more proteins. Iron from the heme group is separated and either bound to proteins and stored in the liver or put right back into a new hemoglobin molecule, and the heme gets turned into bilirubin, a yellowish pigment that goes to the liver where it's added to the bile that it secretes into the intestine and eventually leaves the body in your poop. Now that you know how your erythrocytes function naturally, it's easier to see how they can be messed with, often with bad results. You can dope your blood in a few different ways, but the most common technique is to inject natural or synthetic EPO hormone to boost your red blood cell production. It's also possible to draw and store some of your own blood and then transfuse it back into your body after it's recovered from the blood loss, effectively raising your volume of red blood cells. The logic, if you can call it that, is that more red blood cells equals more oxygen being carried to your muscles and therefore better physical performance. Now, the extra oxygen can't change your actual muscle strength, but the added aerobic capacity does reduce muscle fatigue and enhance endurance by allowing your muscles to work harder for longer. And it can provide enough of an extra edge to win a race, like say at the Tour de France. 7 times. But not only is it banned in athletic competitions, blood doping is also dangerous, because remember, a red blood cell count that's too high thickens the blood, and that actually makes it harder for the heart to pump blood around the body. In addition to defeating the purpose of enhancing the blood's effectiveness, this can lead to blood clots and strokes and heart failure, so no thank you. Plus, cheating sucks. But today you learned about the structure and function of your erythrocytes and of hemoglobin, which they use to carry oxygen. We also went through the formation and life cycle of a red blood cell and studied how their levels are regulated by EPO and their signaling molecules. Finally, we learned how doping the blood is a recipe for disaster and for finding yourself on Oprah and apologizing to everyone you know and losing all of your yellow jerseys. Thanks to all of our Patreon patrons.
Okay, so now we are going to move on to the next one. Non-stop, eh? So we already learned some of the lymphatic system. Now they are going to ask for the immune system. Okay, let me see, muscular lymphatic immune system. Okay, let us see. Body defense. We did this one. Why they want this one again?
Yes, ma'am. They do it again in the unit 13. The same like unit 11.
Yeah.
Okay, so you already learned all those things. Let me see what the new has there. Is there anything new or the same?
No,
It's the
ma'am. All
same.
the
I think they just
same.
want to link that function of the lymphatic system being an immune system. I think they just want to link it. So if we've done it, we'll just keep.
Yeah,
Yeah,
right.
no, that's I'm checking whether we did all the slides or not. That's I'm just going
Yes,
to
we
check.
did. We did all the
All
slides.
of them? All of them?
Yes.
Wow. So why they are doing this again?
I
They're
think they
linking
made
it.
a mistake.
Okay, that I have to write it down then. Chapter 12 is the repetition.
Chapter 11. Unit 11.
No, it said the chapter 12, lymphatic system and body defense.
Oh, yeah, yes, ma'am.
Lymphatic system and body defense. Repeat.
Unit 13 is the immune. Repeat.
Yeah. Yeah.
Okay, so let me see. This is the lymphatic system, innate, everything is there. Okay, let me see. Cancer cells, swelling, phagocytis, innate defense system, redness, it's swelling. I don't like to miss anything, then lysosome, innate, non-specific immunity, all of them we have done it. Okay, so this one is done. So then next is the defense. What is the defense? This is the 12. And
13.
that is the 13 we did.
Yeah.
That is the same, right? Why is called
Yeah.
12? It's the 13. Yeah, this is the chapter 13, not
Yes.
okay.
Go to
So
14.
yeah, I'm just like to see the video crash course is also same.
Oh, yeah.
Same, right?
I don't know. I'm not sure.
Just let us open one and see.
So as I was saying, ever since I switched to Lecturio, I've felt empowered, like I can study anytime.
You may not know it, but your body is engaged in a never-ending battle. You are literally
video
covered in
is
staph
for
and
immune
strep and E.
system.
coli and all sorts of dubious characters that are intent on using you and your body's many resources to feed themselves, find shelter, and reproduce as much as they want. And hey, we all gotta make a living, but it is not your job to give these guys a free lunch. So your body has developed a 3-part policy toward these shady customers, and its enforcement is handled by your immune system. The
You
immune
did
system
it,
is
right?
different from all the other systems we've
You have watched this video, right? Hello?
Don't
No,
recall.
we did not.
No, ma'am, we didn't watch that yet because that's the part of immune system.
Okay,
talk about
so
this here, and that it's
okay, so basically the all PowerPoints are same. Okay, that is nothing new. So what's the video? Okay.
Okay, thank you.
Okay.
Yes.
not a specific tissue, organ, system kind of system. Instead, it involves a whole bunch of different tissue groups, organ systems, and specialized but widely distributed defense cells. Together, this league of extraordinary substances joins forces to perform all of the defense functions your body depends on to keep you alive in an incredibly germy world. And the first line of defense in this never-ending battle, that's your innate or non-specific defense system. Like your average frontline soldier, it's prepared to immediately engage with anyone suspicious, and it mostly includes stuff we were born with, like the external barricades of your skin and mucous membranes, and internal defenses like phagocytes, antimicrobial proteins, and other attack cells. But some enemies must be fought with special forces, and here your body can deploy your adaptive or specific defense system, which is more like your SEAL Team 6. It takes more time to call in, but it's specially designed to go after specific targets, and it keeps files on those bad guys so it knows how to handle them next time around. But today we're going to focus on your innate system and look at how it uses an arsenal of physical and chemical barriers, killer cells, and even fever to keep you healthy, proving that sometimes the symptoms we associate with illness are actually the signs that we're healing. Just because something is simple doesn't mean that it can't be elegant. I mean, your body is capable of some incredibly sophisticated things, including defending itself from infection. But occasionally, there's something to be said for brute force. And a lot of your innate immune system's functions aren't exactly subtle. For example, your body's very first line of defense is a simple physical barrier. And it works. Like a wall around a fortress, your skin does a fantastic job of keeping out all manner of malevolent microorganisms. As long as that tough, keratinized epithelial membrane doesn't get torn open or busted up too much, you could probably, like, make snowballs out of raw sewage and still be all right. Although, no. Your many mucous membranes also provide a handy physical barrier. You'll remember that they align any cavity that opens up into the germy outside world, including the respiratory, digestive, urinary, and reproductive tracts. Not only do your skin and mucosa supply simple physical protection, they also pack some serious chemical weaponry. Eat some questionable leftovers for lunch? Don't worry, your stomach is literally filled with acid, so you probably are covered. Walk face first into your coworker's nasty sneeze cloud? No worries, your nasal passages can whip up a tissue box worth of sticky mucus to help trap viruses before they enter your lungs. You've also got bacteria-fighting enzymes in your saliva and lacrimal eye fluid, and peptides called defensins in your skin and membranes that help keep bacteria and fungi from setting up shop around inflamed or scraped skin. Which, no matter how careful you are, you're gonna get one way or another. Maybe you shave with a dull blade, or you just brush your teeth too hard, and don't get me started about the dangers of bagel cutting. So when you've breached that first simple line of defense, it's time to call on your second line of internal innate defenses. This is where your body starts pulling strategic maneuvers like firing up a fever, releasing chemical signals, causing inflammation, or other defensive tactics that help identify and attack infectious invaders. Some of the first defensive cells on the scene are your phagocytes. Their name literally means to eat, and like Pac-Man, they indiscriminately chase down intruders and gobble them up. And and they come in a few different varieties. First, you got neutrophils, which are the most abundant type of your white blood cells. They kind of self-destruct after devouring a pathogen, and in fact, you've probably seen piles of their little dead bodies, because that's what pus is made of. But the bigger, tougher phagocytes are the macrophages. They're derived from monocyte white blood cells that have moved out of the bloodstream to occupy tissues, and some are free types that patrol tissues looking for creepers, while others are fixed, attached to fibers in specific organs, devouring anything suspicious that passes by. So when a macrophage, in say the finger I just cut slicing a bagel, sees a new bacterium coming along, it snares it using cytoplasmic extensions, reels it in, completely engulfs it, and essentially digests it and spits the rest out. And unlike neutrophils, it can do this over and over again, like a boss. But not all your defense cells are phagocytic. You've also got cells with what is by far the awesomest name of any cell in the body, the natural killer cells. You can call them NK cells if you want to, but like, why would you do that? Anyway, these tiny assassins patrol your blood and lymph looking for abnormal cells and are unique in that they can kill your own cells if they are infected with viruses or have become cancerous. How can they tell? A normal, healthy human cell contains a special protein on its surface called MHC1, or major histocompatibility complex. But if it's infected, it stops making that protein. And if an NK cell detects a defective cell, it doesn't swallow it whole like a macrophage. It pokes it with an enzyme that triggers apoptosis, or programmed cell death, which is pretty awesome. So those are some ways your innate immune cells handle their enemies, but how do they know where to look in the first place? So let's talk strategery. So say you're in a banana factory and you slip on a banana peel and you scrape your knees. Your outer fortress has been breached and the pathogens are just flooding in like orcs through Helm's Deep. Banana factories are very dirty places. Now your body wants to contain the spread of pathogens, clean up the mess, and get healing as quickly as possible, so it cues up your inflammatory response. This is basically an internal fire alarm, only it uses chemicals instead of sirens to get the message across, and instead of smoke and fire, you sense redness, swelling, heat, and pain. For example, in the event of injury, specialized mast cells in your connective tissue send out histamine molecules, and histamine is great at calling in the cavalry. For one thing, it causes vasodilation, which creates redness and heat at the site of the injury. Now, those things might freak you out a little, but they're actually signs of healing. The increased temperature, for example, ratchets up the cell's metabolic rate so they could repair themselves faster. Meanwhile, histamines and other inflammatory chemicals also increase the permeability of blood vessels, causing nearby capillaries to release protein-rich fluids. This causes swelling, which again is actually a good thing because that leaked protein helps clot blood and form scabs, while the lymphatic system sucks up and filters that extra fluid, cleaning it up before putting it back into your bloodstream. And of course, like chum to sharks, an inflamed knee is also going to attract a bunch of local phagocytes, which find it easier to escape your now leaky capillaries, and lymphocytes that are also flowing freely, helping to destroy pathogens while also cleaning up dead cell wreckage. And don't forget, during all this, the neutrophils have been doing their best, but they were the first wave to arrive, so by this time, they're starting to die in heaps. They're triggered when the injured knee skin cells release chemicals that begin leukocytosis, the release of neutrophils from the bone marrow, where they're made, into the bloodstream. To attract the neutrophils to the damaged area, inflamed endothelial cells in the capillaries send out chemicals that act like homing devices. And when the neutrophils arrive, they cling to the capillary walls near the injury, flatten themselves out, and squeeze through the vessel walls to get to work. Your big monocytes eventually roll up to the battle and transform into hungry macrophages, replacing the first line of now dead neutrophils and basically just eating up any lingering enemies and then cleaning up the carnage. Now, all this works pretty well in most circumstances, but you may have noticed with a more major injury or an especially nasty virus or infection, sometimes your local troops get overrun. When white blood cells and macrophages run into more foreign invaders than they can handle, they let loose pyrogen chemicals that tap the hypothalamus and raise your body's thermostat, calling in a systemic fever to burn everything. The resulting temperature rise increases the metabolism of your cells so they can heal faster, and it also tells the liver and spleen to hold on to all of their iron and zinc so those things can't contribute to bacterial growth. But even then, sometimes you find yourself facing a more formidable foe. That's when you call in the specialists, your adaptive immune defenses. And to learn exactly how they save the day, you have to watch next time. But for now, you learn that your immune system's responses begin with physical barriers like skin and mucous membranes, and when they're not enough, there are your phagocytes, the neutrophils and macrophages. You also learned about natural killer cells and the inflammatory response, and watched as all of these elements saved the day when you slipped on a banana peel. Thank you to our headmaster of learning, Lenaya Boyev.
How white do you think your teeth really are? Let's try the tissue test. Ugh, still yellow.
There's toothpaste white and there's Crest 3D White Strips White. They whiten like a $300 professional treatment. And to maintain your brightest smile, Try Crest 3D White toothpaste.
They're coming straight in. Trust me, I've got nothing.
What's true in World of Warcraft is also true in your immune system. To defeat your enemy, you have to know your enemy, uncover its weaknesses, learn how to see it before it sees you. We've already talked about how your innate defense system keeps out or quietly neutralizes pathogens without too much fuss. But sooner or later, a threat's gonna come along that's stronger than what the first responders can handle. That's when it's time for the adaptive or acquired immune system to step in. While your innate system takes its zero-tolerance policy very seriously and tries to toast any foreign microbe that it encounters, your adaptive system does things differently. It has to be expressly introduced to a specific pathogen and recognize it as a threat before it will attack. As its name suggests, you're not born with a working adaptive immune system. It's slow to act, in part because it takes time for it to shake hands with so many pathogens and get to know them. These introductions may be organic, like touching a dirty faucet in the bathroom or walking into a sneeze cloud, or they may be premeditated, which is why vaccination is pretty much the greatest thing to happen to medicine ever. But once it's been introduced to a potential threat, your adaptive defenses never forget it. And this ability to remember specific pathogens is one of the key differences between the adaptive and innate defenses. Another main difference is that adaptive immunity is systemic. Rather than being restricted to a particular infection in, say, a sinus or a sliced finger, your adaptive system can fight throughout your whole body at once. And it does this by deploying one or both of its separate but cooperating defenses, your humoral immunity and your cellular defenses. Your humoral immunity, which you might not have heard of before, works by dispatching important proteins that I'm sure you have heard of, antibodies. They're made by special white blood cells, and they patrol the body's humors, or fluids like blood and lymph, where they combat viruses and bacteria moving around the interstitial space between your cells. Much of what you know or have heard about or think of when your immune system comes up actually has to do with your humoral immunity. It's why if you had mumps as a kid, you probably don't have to worry about getting it again for the rest of your life. It's also why doctors and nurses and patients who have been infected with the Ebola virus, a disease once thought to be incurable, have lived to tell about it. And it's why vaccinations work. Whether you're protecting yourself from infections or playing an MMO, one of the first steps in any good defensive strategy is to be able to tell your friend from your foe. And in the case of your immune system, that means being able to identify antigens. An antigen could be an invader from the outside world, like a bacterium, virus, or fungus, or it could be a toxin or a diseased cell within your own body. But in any case, antigens are large signaling molecules not normally found in the body, and they act as flags that get the adaptive immune system riled up. So let's say a flu virus gets inside of you, and it's floating around trying to find a good host cell to start multiplying inside of. Before it finds that cell, hopefully it will be paid a visit by one of the stars of your humoral response, a B lymphocyte. Like all blood cells, these guys originate in your bone marrow, but unlike other white blood cells, they also mature in the bone marrow too. And as a B cell matures, it develops the ability to determine friend from foe, developing both immunocompetence, or how to recognize and bind to a particular antigen, as well as self-tolerance, or knowing how to not attack your body's own cells. Once it's fully mature, a B lymphocyte displays at least 10,000 special protein receptors on its surface. These are its membrane-bound antibodies. All B lymphocytes have them, but the cool thing is every individual lymphocyte has its own unique antibodies, each of which is ready to identify and bind to a particular kind of antigen. That means that with all of your B lymphocytes together, it's like having 2 billion keys on your immune system's keychain, each of which can only open one door. So part of your immune system's strategy is just to win with overwhelming odds. The more unique antibodies your lymphocytes have, the more likely it is that one will eventually find, bind to, and mark a particular antigen. Once they've matured, B cells colonize or seed your secondary lymphoid organs like your lymph nodes and start roaming around in your blood and lymph. At this point, they're still naive and untested, and they won't be truly activated until they meet their perfect enemy match. Which brings us back to the flu virus. When the right B cell finally bumps into an antigen it has antibodies for, usually in a lymph node or in the spleen, and recognizes it, it binds to it. This summons the full power of the humoral immune response, and the cell basically goes into berserker mode. Once activated, the B cell starts cloning itself like crazy, quickly producing an army of similar cells, all with the instructions for the exact same antibodies that are designed to fight that one one particular antigen. Most of these clones become active fighters or effector cells, but a few become long-lived memory cells that preserve the genetic code for that specific successful antibody. This ensures that if and when the antigen returns, there will be a prepared secondary immune response that's both stronger and faster than the first. This is key to why vaccinations are so brilliant and important, which I'll come back to in a minute. But while the memory cells are just there to hang back and record things, the effector, or plasma cells, are packed with extra amounts of rough endoplasmic reticulum, which acts as an antibody factory. These cells can mass produce the same antibodies over and over for that particular invader, spitting them out into the humor at a rate of around 2,000 antibodies per second for 4 or 5 days until they die. And the antibodies they make work the same way that the membrane-bound ones do. They're just free-floating. So they ride the tides of blood and lymph, binding to all the antigens they can find and marking them for death. Now, antibodies can't really do the killing themselves, but they do have a few moves that could make it hard for intruders to take hold. One of their most effective and common strategies is neutralization, where antibodies physically block the binding sites on viruses or bacterial toxins so they can't hook up to your tissues. And because antibodies have more than one binding site, they can bind to multiple antigens at the same time in a process called agglutination. The resulting clumps can't get around easily, which makes it easier for macrophages to come and gobble them up. And not only that, but while all this is going on, antibodies are also ringing a chemical dinner bell, calling in phagocytes from the innate immune system and special lymphocytes from the adaptive system to destroy these messy little antigen antibody clumps. So the point of all of this in the short term is to keep you healthy, but in the long term, this process also adds to your overall immunity. The humoral response allows your body to achieve immunity by encountering pathogens either randomly or on purpose. Active humoral immunity is what we were just talking about. It's when B cells bump into antigens and start cranking out antibodies. This can occur naturally, like when you catch the flu or get chicken pox or pick up some nasty bacterial infection, or it can happen artificially, particularly through vaccination. Most vaccines are made of a dead or extremely weakened pathogen, and they work on the premise that because a secondary immune response is more intense than a primary response, by introducing a pathogen into your body, you're priming it to fight hard and fast should that antigen show up again. In the case of typically non-fatal infections like the common flu, this immunity should at least spare you from some of the most severe symptoms. But in the case of more serious diseases like polio, smallpox, measles, and whooping cough, vaccinations can be truly life-saving. Now, some antigens, like those for mumps or measles, don't really change much over time, so a few immunizations will leave you set for life. But others, like influenza, are constantly evolving and changing their surface antigens, so immunity to last year's flu probably doesn't work against this year's flu. Still, acquired immunity doesn't have to be active. Babies, for example, naturally obtain passive humoral immunity while still in the womb. They receive ready-made antibodies from their mothers through the placenta and later on through breast milk. And that works pretty well for a few months, but the protection is temporary because passively obtained antibodies don't live long in their new body, and they can't produce effector cells or memory cells, so a baby's own system won't remember an antigen if it gets infected again. You can also acquire this kind of temporary passive immunity artificially by receiving exogenous antibodies from the plasma of an immune donor. This is what recently saved some doctors and nurses who had contracted the Ebola virus from infected patients. A serum was made from the blood plasma of other medical workers who had been infected and survived. The antibodies helped defend the patients from the virus before their own active immunity could identify that particular antigen and start creating their own antibodies. It's not the same as a vaccine, which immediately engages your B cells, but it can buy a patient some crucial, life-saving time against an infection that would otherwise quickly kill. But B cells and antibodies are only part of the immunity equation. There are plenty of pathogens that quickly worm their way right inside your cells, where they're safer from the humoral response and free to multiply as much as they'd like. Luckily, your immune system has yet another game plan and a new set of players ready to fight that final battle with cell-to-cell combat. Make sure you catch our final episode next week and learn all about this epic battle royale. But as for today, in our second to last episode, you learned how the adaptive immune system's humoral response guards your extracellular terrain against pathogens. We looked at how B cells mature, identify antigens, and make antibodies, and how antibodies swarm pathogens and mark them for death. We also talked about active and passive humoral immunity and how vaccines work. Thank you to our headmaster of learning, Linnea Boyev, and thank you to
Paramount Plus is now Canada's
Come on. Let us search the respiratory system, okay, today. So what is the respiratory system? Through which we take the respiration. That means oxygen, we inhale oxygen and exhale carbon dioxide. What are the different parts of the respiratory system? Nose. Then after nose, the tube is going down near the throat region. That is the pharynx. After pharynx, there is one structure. It's a tube. That part is called the larynx, which holds the voice box. Then we have the trachea. Then trachea, then branches to bronchi, and then bronchi, left and right bronchi, and then this is going to more branch and branches. So it's called the bronchi, then bronchus, then bronchi, then lungs, and from the lungs that is alveoli. So this is our nasal cavity. This is the nostril. You are taking the blood. Sorry, you take taking the inhale oxygen. Then it's going through here. This is the pharynx. It passed through the pharynx. Then the oral cavity pharynx. After pharynx, here is the voice box. It's called the larynx. Larynx is continuing as the trachea, and this trachea then divided into left and right trachea. So now we have to, so this is, and they are only just on this one, and then trachea, then after trachea is called the bronchi, when it's going to be divided in two before entering the lungs, bronchi, and then bronchi is going to be divided, it's like the small branches called bronchiole, and ultimately it ends up within a alveoli. So what are the main function of the respiratory system? Gas exchange between blood and external environment in the lungs, alveoli. So what I told, the gas exchange mainly takes place in the alveoli of the lungs. Passageways to the lungs purify, humidify, and warm the incoming air. When you are inhaling, the air is going from the outside, safe for when you are taking the air during the cold season, -20, the air which is entering in your nose, nostril, and going is very cold. So all the structures, pharynx, larynx, trachea, they helps even the purify so that they trap any bacteria or anything. They are mucous membrane. And then they make it when it's passing through body temperature is high, so make the air warm as much as possible through their trach. Okay, so passages to the lungs purify, humidify, and warm in the incoming air. What is the function of the nose? Nose, only external visible part of the respiratory system. Only you can see the nose. All other inside. So only the exterior visible part of the respiratory system is nose. So air enters the nose through the external nostrils, which is the name called the nares. The interior of the nose consists of a nasal cavity divided by a nasal septum. We are going to see the structure. So here is a nostril, is going there. You doesn't need to know all of the things, but it's important that air is going through the nostril, then passing through the pharynx, then to the larynx, then to the trachea to the alveoli. But what is here? It is divided by a palate. Why? Because we have the oral opening and we have the nasal opening, right? So if we doesn't have the separation, what is going to happen? All the time your food or saliva and everything is getting mixed with the nasal cavity and mouth cavity. And that is not good. So this is getting too. So this acts as a separator. It's called the septum, heart palate. Okay. And here we have the, we doesn't need to know all of the things. It's too much bone, this, that. So we have three parts. In the nasopharynx, we have the pharyngeal tonsil. tonsil is located here. Okay, pharyngeal tonsil. We have another tonsil, is called the oropharynx, which is just beneath the heart palate. It is called as palatine tonsil. And you have another below, is called the lingual tonsil. And we have the flap-like structure at the back of our septum, which governs the food and respiration air entry. When you are eating the food, it blocks the nasal opening. And when you are respire, it blocks the food. So that's why they cannot be mixed. You cannot do eating and respiration at the same time. Even you do, it's not going to work for the who is eating that one. That time it's going to only block the valves of the respiratory system and digestive system and allow the air to come in. So anatomy of the nasal cavity, olfactory receptors are located in the mucosa of the superior surface. Olfactory receptors means which helps you to smell the different things. What is the smell? Is the smell of a perfume or poo or pee or grass or flower or anything. So that is the olfactory receptors are present in your mucosa of the super surface. The rest of the cavity is lined with respiratory mucosa that moisten the air and trap incoming foreign particles. So we have the olfactory receptors fairly from the beginning to almost end, but at the same time, this rest of the cavity is lined with the respiratory mucosa. That what is the function? It moistens the air and trap incoming foreign materials. And then the lateral walls are projections called conchi. This is important. What is the lateral walls of nostrils has? It's called the conchi. What is the function of the conchi? Is increase the surface area. These are the lateral walls projection. It helps to pass and purify and moisten the air when it's coming in. So lateral walls are projections called conchi. It increases the surface area, increase air turbulence with nasal cavity. And then the nasal cavity is separated from the oral cavity by the palate. We call the anterior palate bone and posterior palate muscle. So this one. This is the bony palate and this is the muscle palate. Muscle palate is extended as a uvula and this is the bony palate. So mouth cavity is separated by the nasal cavity. Which one did this one before? Okay. Conchi, okay. Increase surface area and increase theater. And this one also did. We got the hard palate and soft palate. So paranasal sinus, what are those? This is the cavities with bones. When we did the skeletal structure, we told about that. Cavities with bones surrounding the nasal cavity called sinuses. Sinuses are located in the following bones: frontal bones, sphenoid bone, ethmoid bone, maxillary bone. What is the sinuses? This is giving the. So these are the different bones. Is said. It just only showed you the frontal sinus here and sphenoidal sinus here, not all of them. So just remember they have 4 sinuses. That's all. You doesn't need many. And each bone, you doesn't need that. So you have the 4 sinuses. So what are the function of the sinus? We already learned in our skeletal system. It lightens the skull, right? Because it has a sinus. Because it acts as a resonance chamber for speech. In the sinus, when the air passes through the larynx and this empty spaces, that is the sinuses, helps to resonate so that we can put the speech and produce mucus that drains into the nasal cavity. Also, that's why when you get the cold infection, then you can, the fluid what is have is become thickened, infection cause the mucus, and then you feel the pressure, sinusitis, and other things. So produce mucus that drains into the nasal cavity. What is pharynx? Pharynx is the muscular part. This is the muscular passage from nasal cavity to the larynx. So from the now nasal cavity, air is coming. These muscles help to move the air inside. So we have three regions of the pharynx: nasopharynx, oropharynx, and laryngopharynx. So nasopharynx is superior region behind the nasal cavity immediately. Oropharynx is the middle region behind the mouth, and laryngopharynx is inferior region attached to the larynx. So it's from the top to down. So oropharynx and laryngopharynx are the common passages for the air and food. That is the main things in the pharynx. So see, this is the hard palate. These are called the nasal conchae. This is the folding of the lateral wall of the nostril, nose, and this increases the surface area so that they can moisten the air, trap the any bacteria or anything, filter, and send the air to the down in the pharynx. So this from here, from the down, is called the pharynx. We have three pharynx. One is nasopharynx, which is just behind the nostril aperture where it's making connection. Then we have the oropharynx, which is just behind the mouth where we have the oral cavity opening, and we have the laryngopharynx, which is going to the larynx. So they have three of them. So pharynx has three parts: nasopharynx, oropharynx, and laryngopharynx. So now the nasopharynx is up, is getting blood here, air here. But here, the junction, here, the oropharynx and laryngopharynx, from here, both foot, here, when the air is coming, there is no problem because it's separated totally. But in this region, oropharynx, and laryngopharynx, in this area, is a passageway for the food as well as the passageway for the air. So whenever you are doing respiration, there is a flap. It's called the uvula. This is going to close this one so that the air can go straight to the lungs. When you are not breathing or you feel hungry, so whenever you start chewing or something, there's this flap-like structure is going to close this one. And when it's closed, the oropharynx and laryngopharynx, if there's food or anything, they can pass through the food one. Okay. What are the tonsils of the pharynx? And tonsils, one of the immune, takes care of the immune system. They trap the bacteria. The first, when it enters to the, especially from the mouth as well as from the air inhalation, the tonsils first trap any bacteria or anything. That's why we get the tonsillitis or the infection of the tonsil very fast, because it is trapped a huge amount of any pathogen or any foreign substances. So there are a couple of nostrils, which is pharyngeal tonsil, is called the adenoid, then nasopharynx. Palatine tonsils are located in the oropharynx, and lingual tonsils are found at the base of the tongue. So these are the different tonsils that I showed you. So this is the palatine tonsil, is near the oropharynx. Then we have the nasopharynx, pharyngeal tonsil, which is just above, just behind the nasal opening. And we have another, uh, what is that one? Nasopharynx, pharyngeal tonsil, palatine tonsil, and lingual tonsil. It is just behind the, just above the larynx. What is the larynx? Larynx is the voice box. It routes air and food into the proper channels. It plays a role in speech and made up 8 rigid hyaline cartilage and a spoon-shaped flap of elastic cartilage. You have to remember the larynx is made up of hyaline cartilage, 8 rigid hyaline cartilage. And it's main the voice box, and it has plays a role in the speech. So you doesn't need to need too much of that. So the structure of larynx is a thyroid cartilage. Epiglottis. What is epiglottis? Protects the superior opening of the larynx so that it can routes the food to esophagus and air towards the trachea. So when swallowing, the epiglottis rises and forms a lid over the opening of the larynx. Vocal folds. We have in that area of the larynx, we have the folds so that the air can pass and resonance so that we can produce the different types of speech. You can sing, you can recite whatever maybe. So vibrate with the expel air to create sound. What is glottis? Between mouth and vocal cords. That's straightforward. What is the name of the trachea? Trachea is the windpipe. So it's generally approximate 4-inch long, and it connects with the bronchi and larynx. So from larynx, it's coming down and going to the trachea before it divides into 2 bronchi before entering the lung, left and right bronchi. It is lined with ciliated mucosa, and it beats continuously in the opposite direction of incoming air. So that in the opposite direction, so that any expel, any mucosa or anything is not going inside the lung or inside the circulatory system. It's beating upwards so that any problem or any infection or anything can be come out. That's fine. It doesn't need to need this one. Primary bronchi. So this is the division of the trachea. Enters the lung at the helium. Helium is any depression, like kidney, you are going to see the structure. So where is this helium? So you can the depression, when we read the kidney, you see there is a depression where the main kidney ducts are entering. So when the depression point from where something enters is called the helium. So it's the helium, this trachea is going to enter in the right and left bronchi in the helium region. So right go to the right bronchus and left go to the left bronchus. So bronchi subdivide into smaller and smaller branches. Once they enter in the lung, it divide and divide and divide. And then it ultimately turns out to the lung spongy tissue is the alveoli. So these are all the structure, doesn't need that. So then what is the lungs? It's a spongy tissue. This is occupy most of the thoracic cavity. What is in front of the lung or back of the lung?
Behind the lungs in the middle?
No, it's front of the lungs.
Of the
Pardon?
lungs. Is in front of the lungs.
Yeah, yeah. Okay. So heart occupies a central position called mediastinum. So heart in middle and lungs starting from the top as the wider and then is getting pointed. Is pointed down here. Okay, and heart is here. Yeah, and that is the thoracic cavity. Okay. So this is important. Okay, heart apex is near the clavicle and superior position. That is the heart. Okay. So oh my God, who is hearing the music? So it's telling that lungs apex is near the clavicle. So this is the clavicle bone, right? Collarbone. So lungs apex is in the clavicle and base is near the diaphragm. So you can see the diaphragm is a diaphragm. So heart apex is where? Which side? Up or down?
Up.
No.
Oh, down, down.
That the pointed portion is called the apex. So it's heart apex is this side, and base is on the right shoulder. Okay, it's in the little bit in this position. It's not straight, but lungs apex is in the clavicle, and lungs base is on the diaphragm. It's rests over the diaphragm. So, and each lung then divided into lobes or fissure. This is important. These questions may come. How many lobes has left lung and how many lobes has right lung? Right lung has 3 lobes and left lung has 2 lobes. Important question. See, left lung has 2 lobes, 1 and 2, and right lung has 3 lobes, 1, 2, and 3. I don't know why. Rasvi, that is a structure. We doesn't need to know so many things. Doesn't need that. So bronchial respiration, as we learned that with the first trachea divided into bronchi, and then bronchi is divided into branches and branches and branches. What is called primary bronchi, secondary bronchi, tertiary bronchi, and then ultimately it forms a bronchioles and then terminal bronchioles. So minute. It looks like a bronchi, but ultimately turns into bronchioles and terminal bronchioles. See, like this one. See, this is the first trachea. From trachea is a bronchi, then it's dividing the primary, then secondary, then tertiary. So it's branches, branches, branches all over the round, and then it's going to end it up in a bronchiole. So this is become enlarged. So this is the terminal bronchiole. From after that, which is only alveolar duct and sac. Mainly the alveolar sacs is there for the gaseous exchange. Now, what is the respiratory zone? Respiratory bronchioles. It has the alveolar ducts. Alveoli is the air sacs. These are the alveoli, air sacs. So this is coming from the spongy tissue of the lungs, the alveoli ducts, and here the exchange takes place. So lungs are the spongy tissue, and the trachea, bronchiole is making the branches. Then lungs, alveoli or alveolar, mainly the gas exchange takes place in the alveoli. That's all. Not to do too much complicated things. How the gas exchange is seen by the process of diffusion, because it's against a concentration gradient and it has a membrane. So oxygen enters the blood and carbon dioxide enters the alveoli, right? Because oxygen from the outside is entering into the blood, and carbon dioxide, which is already in the blood, it has to get rid of. So how is going to get rid of the alveoli? And then alveolar macrophages, there are everywhere there's a blood cell monocyte which acts as the macrophage later. And if there's any blood, something, any bacteria or carbon dioxide, immediately in the normal condition, they like to pick it up so that the person doesn't become sick. So what are the 4 events of respiration? Pulmonary ventilation, moving air in and out of the lung, called breathing. In and out. External respiration, gas exchange between pulmonary blood and alveoli. That is the gas exchange is called the external respiration. Pulmonary ventilation, moving air in and out of the lungs, in and out from the lungs is called the breathing. And external respiration, gas exchange between pulmonary blood and the alveoli. So oxygen is loaded in blood and carbon dioxide is unloaded from the blood. So first you have the respiratory gas transport, then the internal respiration. It says the respiration is a mechanical process because you are inhaling and outhailing, right? So basically this is the pulmonary ventilation. You are inhale and outhale. Inhale is a mechanical process. And how the volume change depends upon the pressure changes. How much air is going to take in and how much air is going to come out. So there are two things, two phases, inhalation, inspiration and expiration.
Miss
Hello,
Moon
ma'am,
Moon?
where are you?
I think she's not in the team. Oh.
Oh.
Maybe she got cut off. Yeah, I think her video dropped.
maybe.
Yeah.
You guys can hear me?
Yes.
Yes,
You got
we
dropped off.
can hear you.
You're back, ma'am.
Huh?
You're back.
back
You've
now.
gone for a while.
How long? I don't know. I didn't do anything. Something is going on. From which slide you didn't able to hear me? I'm just speaking like stupid. Which slide?
We're on the exploration. We finished that one. So the next one would be the pressure. Let me just see. Slide figure 13.8. I think that's where we are now.
We can't see your screen either.
How it happens?
I don't know. That's what happened in the tests and exams too, you know.
Because suddenly, because I didn't change anything, I wanted the PowerPoint only, right? And suddenly I got the call from Saban. I said, what? Why Saban is calling me?
Yeah.
Okay.
It was a good break, so don't worry. Thanks.
Okay, so which one?
It's right there. That's where we
Pressure
are.
differences in the thoracic cavity. Yeah, that one.
This one, right? Okay.
Yes.
Okay. This you doesn't need to know. Okay. So what is the non-respiratory air movements? Generally, you inhale and exhale. That is the normal one. But sometimes some extra things happens which is associated with the respiration. So what are the cough and sneeze? What that cough and sneeze? Clear lungs or debris. At that time, you are also inhaling some more air. Crying, when you are crying, you are in stress, so you are doing the little bit extra respiration. Laughing, that is also emotional, similar to crying. Hiccup, when you are doing the hiccup, you are inhaling some more air, and at the same time you have to exhale it. Yawn, when you are sleeping, like that. Yeah, I like to do yawning right now. I am so tired. Yeah, but anyway, when you yawn, we get more oxygen inside because body is lethargic. Body needs more oxygen to make you wake up. So what are the different? Oh, this one I told you, this is important. What is how much air you can take in, or the bear moves per breath? 500 ml. Okay, 500 ml. In a normal breathing, one person can take, or air can move 500 ml air in the lungs. This is called the tidal volume. This is tidal volume is the amount of air can moves in normal breathing, and that is 500 ml. Which factors can alter this 500 ml? It may be not everybody has exactly 500 ml. Somebody has 450, somebody may has 550, somebody has 700. But which factors alter this? Person size. If you are small, medium, large size, like our dress. Sex. Male can inhale more than the female. So sex, age, younger, or the young generations, or the child, they have more breathing rate faster, they inhale and exhale more. As you aged, your breathing becomes slowed down. So that time you may have little bit less air intake inside your body. So tidal volume may be less than 500. And physical condition. If you are sleeping, the intake and out is not, is normal, is not going to be slow. But you are doing exercising or some other physical condition, depending upon that, the respiration, tidal volume or respiration rate may be different. Okay. So what is the respiratory reserve volume? Your tidal volume is 500 ml, that is the normal. Inspiratory reserve volume, how much you can take with force over the tidal volume. Tidal volume is already 500 ml, and over that, how much you take when you need it, when you are doing vigorous exercise, or you are in the, sometimes you are going up somewhere, you need more air. So that time maximum you can 3100 ml over the tidal volume. And expiratory reserve volume, that you can forcibly exhale. So if you take 500 ml, in theory, you will be able to exhale in normal condition 500 ml. But when you expire the extra air, you can forcibly exhale 1200 ml. So you can take more breath in, but you cannot expel the whole air at the same time. Something is going to stay inside the lung. So this is very important. What is the residual volume? Because even you are taking every breath, you cannot expel out the whole amount of air. There is always some air is staying inside the lung. Otherwise, lung is going to be collapsed. Right? So about 1200 ml of air always stays in normal condition in the lung after your exhalation or expiration. So that is called the residue. That is the leftover. That is called the residual volume. So main is tidal volume, how much you can take in normal breath. The residual volume, which is staying inside the lung even after the expiration, normal expiration, that is the residual volume, and in between there is the IRV and ERV. So forcibly inhale, forcibly exhale. What is the total vital capacity? Is the sum of everything: tidal volume, IRV and ERV. That's all. You doesn't need to the disk space, volume and other things. This you doesn't need it. We are not going to check with the statho like the doctor. So what is the external respiration? Oxygen loaded into the blood. Oxygen go inside, then it's going to the loaded in the blood. And then alveoli has always more oxygen than the blood, because alveoli is part of the lungs, right? And is coming, we are taking the air from the outside, and that air is going to go in the lungs, in the spongy tissue through the bronchi. So alveoli always have more oxygen, and blood has more carbon dioxide. So what happens? The diffusion. Oxygen is higher in alveoli, inside the blood it is less. So oxygen by diffusion moves from the alveoli to the lower concentration area. What is that? Inside the blood. And the same thing happened. The carbon dioxide is more in blood and less in the alveoli. So carbon dioxide moves to the lower concentration, that is the alveoli, to go out. So pulmonary capillary blood gains oxygen. That's the same thing I told you. So blood leaving the lungs is oxygen rich and carbon dioxide poor. This we already discussed. Oxygen travels attached to the hemoglobin and forms oxyhemoglobin. Oh, doesn't need too much. And what is the internal respiration? That when we did the mitochondria, you remember we told you mitochondria, that is the site for the internal respiration, when the gas exchange between the blood and the body cells. External respiration, alveoli and the blood. Okay, and internal respiration, gas exchange between blood and body cells. So here carbon dioxide diffuses out from the tissue to the blood, called loading, and oxygen is diffuses from blood into the tissue. It's called unloading. Just that's all. You doesn't remember the terminology loading, unloading. So in the internal respiration, just internal respiration is the gas exchange between blood and body cell. That's it. So what is the neural regulation of respiration? Who controls this respiration? Obviously, some brain. Brain is the master for controlling everything, right? So activity of the respiratory muscles is transmitted to and from the brain by phrenic and intercostal nerves. It's okay. You doesn't need to name the nerves. So what is the neural centers that control the rate and depth of respiration? Are the medulla and pons region. So what medulla does? It says the basic rhythm of breathing, how we are taking the breathing. We are taking 12 to 16 beats per minute. That is the generally normal. So it sets the basic rhythm of breathing and contains a pacemaker called the self-exciting inspiratory center. Doesn't need that. Medulla sets the basic rhythm of breathing and appears to smooth out respiratory rate, pons. If that is respiratory rate is high or low, the pons area helps to smooth out so that it can comes back to the normal level. So medulla is the main center. So this is important. Normal respiratory rates is called the eupnea. Normal is eupnea, 12 to 15 respiration per minute. Hyperapnea is when the respiratory rate are more than 16. It's more than 16 to above. So when your respiratory stays, sometimes you have the hyperapnea. So due to extra oxygen need, hyperapnea. What are the other, except the neural factors, that is the medulla from the brain, what are the, we already talked, what are the other factors which can influence respiratory rate and death? Body temperature, exercise, talking, coughing. I don't understand. Volition means conscious control. Oh, conscious control when you are doing by intentionally. And the emotional factors. This is, we know, oxygen and carbon dioxide. What is hyperventilation? It's called the tachycardia, right? This is important. When you feel the hyperventilation, when there is a decrease of carbon dioxide in the blood, it's called the acidosis. Breathing becomes deeper and more rapid. So what it happens, blows of more oxygen, carbon dioxide to restore normal blood pH. So hyperventilation means not, you need the more carbon dioxide. Result from, sorry, hyperventilation is a more carbon dioxide there. So the accumulation of carbon dioxide is more in the blood. It's called the acidosis. So they need to get rid of the carbon dioxide and get more oxygen. That you are doing hyperventilation, breathing tachycardia, so that you can get more oxygen in and dispose of the carbon dioxide which has been more in the blood for any reason. That is called the hyperventilation. Results from increased carbon dioxide in the blood. Hypoventilation, when the blood becomes alkaline, alkalosis, extremely low or shallow breathing. So when there is a less amount of carbon dioxide in the blood and you need the more carbon dioxide. So that time they told to inhale your own exhale. So it can go inside your body and can raise the carbon dioxide level. Okay, so what is emphysema? Chronic bronchitis is called the emphysema. And this is the major cause of death and disability in the United States. Chronic bronchitis is always the inflammation of the lung non-stop. There are couple of disease which is linked to respiratory disorder. It's called the chronic obstructive pulmonary disease. You probably have no COPD. Who develops the COPD mostly? Who are smoker? Who has the labored breathing for any reason? Who has the too much coughing and frequent pulmonary infection? They can get the COPD. So most victims are hypoxic, retain carbon dioxide and have respiratory acidosis. That means they have more carbon dioxide in the body, inside their body, in the blood. So they are damaging their respiratory system and ultimately develop the chronic obstructive pulmonary disease. Ultimately, they are going to end it with the respiratory failure. You doesn't need to. So lung cancer, another is the disease. It mainly occurs who are chain smokers. That is chain smokers. So there are three common types: squamous cell carcinoma, adenosarcoma, small cell sarcoma. That's I just only know lung cancer is the increased incident is associated with the smoking. Doesn't need the developmental aspects. Cystic fibrosis. This is cystic fibrosis is very important. What is over secretion of thick mucus clogs the respiratory system. When there is over secretion happens in the lung and it clogs all the pores and everything, so there is obstruction of the gaseous exchange. This is the cystic fibrosis. This is important. What are the respiratory rate changes throughout the life? Adults, 12 to 18 respiration per minute. So that's why I said the respiration rate and things is dependent on the age. What age 5 to 15, age 5, they have 25 respiration per minute. The kids respire more. Infants, 30 respiration per minute. And newborns, 40 to 80 respirations per minute. This doesn't need. What is the asthma? Lot of people suffers from the asthma. It's the chronic inflamed hypersensitive bronchial passage and response to irritants with dyspnea, coughing, and sneezing. So it's inflammation in the bronchial passage. That is the asthma. Asthma also comes from the allergy. So as you age, elasticity of the lungs decreases. Obviously, lung capacity will be decreased. Blood oxygen level is decreased. Some stimulating effects of carbon dioxide decrease, and elderly often hypoxic and exhibit sleep apnea because they cannot get the proper respiration at night, and most risk of respiratory tract infection. Done. Okay, so we whatever really important, I just told you guys. I don't think there is we need more than that. Too much. 92 slides. Okay, so let me see what is the crash course here. We just do the respiratory system, right? So we have digestive system for this unit. How many units I have? And how many we have to do exams? 15, 1, 2, 3, 4, 5, 20. Test 4. And then it's going to be the final. Okay. Ooh. Can you hear?
The attraction towards new experiences
Yes,
is
thank
powerful.
you.
Yes, miss.
Okay. Okay.
Alienware gear isn't made to resist.
Let me introduce you to one of the bravest pioneers in the history of life on planet Earth. An organism that blazed the trail for every single vertebrate that lives on land today and many that don't. It's one of your most important ancestors. Meet, well, It doesn't have a name. And we don't exactly know what it looked like either. But we do know that about 380 million years ago, this fishy-looking thing with big fleshy fins achieved one of the animal kingdom's greatest milestones: breathing air. Sounds simple enough, but believe me, it wasn't. Because for billions of years before this fishy ancestor came around, basically all of life evolved in water. From the very beginning, the earliest, simplest forms of life, like bacteria, extracted oxygen they needed right from the water through their membranes. And they did it through simple diffusion, when a material automatically flows from where it is concentrated to where it is less concentrated, so it balances out. Diffusion works really well, and it requires zero effort, but it wasn't going to cut it in the big leagues. Anything larger than a small worm is simply too big and needs too much oxygen for diffusion to work. So in order to get bigger, early life forms needed a circulatory system that could move bulk amounts of oxygen around faster inside their bodies, and a respiratory system to bring more oxygen in contact with their wet membranes. So their respiratory surfaces moved from their outer surfaces to the insides of their bodies. First there were gills, but gills of course still only work inside of water. And a little more than 380 million years ago, this was starting to lose some of its charm. Earth was getting warmer, the seas were getting shallower, and much of the planet's surface water had lower concentrations of oxygen than it used to. Finally, a humble little lobe-finned fish got fed up, swam up to the water's surface, and started breathing air. It could do this because it had evolved a fancy new interface to move gases between the air and its cell membranes. I'm talking about lungs. Wet lungs. With an efficient new way to take in nearly limitless amounts of oxygen from air, animals were eventually able to get bigger and more diverse over the ages, and now all of us lung-having vertebrates share that common ancestor. For lots of animals, including humans, those lungs come with a bunch of other equipment like protective ribs, a stiff trachea, and in mammals, a strong diaphragm. And together, they form your respiratory system, which happens to be best friends and business partners with your circulatory system. It's only by working together and using both the bulk flow and simple diffusion of oxygen that they can make possible the process of cellular respiration. In other words, life itself. So a lot of improvements have been made to it over the eons, but the respiratory system that you are using right now is your inheritance from that ancient, ambitious fish, leader of one of the most important anatomical revolutions of the past 500 million years. Pretend for a minute that you can't breathe. Like, you just don't have lungs anymore. You are some bizarre evolutionary oddity, a huge human-shaped organism that doesn't have a respiratory system. Instead, you get all of your oxygen the way that your oldest, smallest evolutionary ancestors did, by simple diffusion, or at least you try to get all your oxygen that way. How would it work? Well, poorly, and that's partly because one of the keys to efficient diffusion of any material is distance. If you want a molecule to diffuse across a space quickly, you want it to be as close to its destination as possible with the fewest obstacles in the way. But for a single molecule of oxygen to diffuse from the air through, say, your scalp and then go to a neuron deep inside your brain, it would have to move through your skin and then your skull and then your connective tissue and all sorts of things. It would eventually get there, like maybe a month later, but at that point, the cell that needed the oxygen in the first place would have, suffocated to death. Basically, obtaining oxygen through diffusion alone is like wanting to go to a party at your friend's place across town and then walking 20 miles to get there. You could do it, but it would take forever, and by the time you arrived, you'd be all haggard and the party would be over. So diffusion alone isn't enough to get the job done. We do use it, but only when a whole bunch of the materials we need are right up against the tissues that can absorb them. So you know what else we need? Bulk flow. Bulk flow is like public transportation. It moves large numbers of molecules quickly. Rather than walk the whole way across town, you can hop on a bus with a bunch of other people and get there in 20 minutes. Every time you take a deep breath, you bring in about 100 quintillion oxygen molecules into your lungs all at once. They're on a bulk flow bus ride. And once those oxygen molecules filter down into the cells in your lungs, they're suddenly very close to the blood they're trying to reach. All they have to do is diffuse across 4 layers of cell membranes to get from the lung cell into the blood. It's like hopping off the bus and then walking a half a block to your friend's apartment. That's why your respiratory system is the way it is. It's set up to take full advantage of both bulk flow and simple diffusion. The bulk flow part of things is handled by some of your system's biggest and most obvious moving parts, starting with your lungs, which basically operate like a pump or a bellows. They don't have any contractible muscle tissue because they need to be able to expand, so they require outside help in order to move. Enter the diaphragm, a big thin set of muscles that separates your thorax from your abdomen. When your lungs empty, your diaphragm relaxes and looks kind of like an arc pushing up to squish your lungs. You also have the weight of your rib cage pushing on your lungs from the top and sides, and together these forces decrease the volume of your lungs. When you breathe in, your diaphragm contracts, pulling itself flat, and your external intercostal muscles between your ribs contract. They lift the ribs up and out, causing the chest cavity to expand. This makes the pressure inside your lungs lower than the air outside your body, and since fluids like gases move from areas of high pressure to low pressure, the lungs fill up with outside air. Then the diaphragm relaxes again, and the weight of the ribs settles in, and the pressure inside the lungs becomes higher than the outside air, and the air rushes out. And that, my friends, is breathing 101. Now, your respiratory system contains a lot of parts besides your lungs, some prominently displayed on your face, others hidden deep within your chest, and functionally all of these organs fall into one of two physiological zones. The upper parts that funnel the air in make up what's known as the conducting zone, and it starts with this thing. Your nose is supported by bone and cartilage and the bristly hairs and mucus inside it that help filter out dust and other particles. But it, along with your sinuses, performs another important function. It warms and moistens incoming air so it doesn't dry out those sensitive lung cells that must remain wet. Remember, moisture is key. We evolved from organisms that lived in water, so just like with our aquatic bacterial ancestors, we need water for oxygen to dissolve into before it can diffuse across the phospholipid bilayer membrane of our cells. Now, if you've ever choked on a poorly timed sip of water, you've noticed that you breathe through the same tube that you also move foods and liquids through. This is yet another leftover from those first fish lungs, which evolved as a branch off the esophagus. Looking back, it was not ideal, but we are stuck with it. So the stuff that you swallow soon encounters the epiglottis, a little trapdoor of tissue which covers the larynx and directs bites of sandwich and sips of cola towards your esophagus and keeps them out of your lungs. And you'll notice that the esophagus, which heads to your stomach, is nice and flexible, while your trachea, or windpipe, is rigid and has prominent rings. That's because your trachea is basically built like a vacuum hose. Since the lungs create negative pressure with every breath, the trachea needs those rings to keep it open. If it were soft and floppy, it would collapse every time the pressure dropped and you wouldn't be able to breathe. From there, the trachea splits in two, forming the right and left main bronchi. You can imagine these inner lung parts as sort of an upside-down tree. Now we are in the lung tissue and have entered what we call the respiratory zone. This is where the actual gas exchange occurs, and everything you find here has a form to suit that function. So the smaller branches of the upside-down tree are bronchioles, which taper down into progressively narrower tubes until they empty into the alveolar ducts and then dead end into tiny alveolar sacs, where the bulk of the gas exchange finally occurs. Because that's where each sac contains a cluster of alveoli, these tiny cavities lined with super thin wet membranes made of simple squamous epithelium tissue. It's here that oxygen molecules dissolve in the wet mucus, diffuse across the epithelial cells, and then cross the single layer of endothelial cells lining the capillaries to enter the bloodstream. And of course, it's also where carbon dioxide diffuses out of the blood and then follows the same route back up to the nose and mouth where it's exhaled. So it's your alveoli where diffusion meets bulk flow, because while you're picking up oxygen and dispensing with CO2 one molecule at a time, you're doing it in enormous quantities at any given second. Both of your lungs contain about 700 million alveoli, which together provide an amazing 75 square meters of moist membrane surface area. So the principles that make respiration possible are relatively simple: diffusion and bulk flow. And so are the mechanisms in your body that use them. It just took us about 400 million years to figure out how to make it all work. But today you learned how it does work, including the mechanics of both simple diffusion and bulk flow, and the physiology of breathing, and the anatomy of the conducting zone and the respiratory zone of your respiratory system. Thank you to all of our Patreon patrons who helped make Crash Course possible for themselves and
So you want to watch the next one, or you want to watch tomorrow?
Can we do it tomorrow?
Tomorrow? Okay. Okay. Sure. No problem. Okay.
Thank you.
Thank you. So tomorrow we are going to probably do the last one, right? This is the 14 or 13. Okay. Anyway, so Friday we are going to get some time to review, and tomorrow we'll see if we get some time. We will give you some time to review the study. Okay. Okay. And then you want to do the exam on Friday or Monday again?
Ma'am, I want
Friday, miss?
Friday. Remember I told you I'll be going away?
Who is that?
Me, I said I mentioned last week that I won't be here on the 23rd. Sorry, 24th.
Yeah, only thing is that it's going to be too much pressure for you guys. It's up to you. But if you want, you can take the exam, but others can take it on the other day. Or if everybody wants to take it in the Friday, I have no problem because it's going to be
No,
little
I
bit.
won't be here on Friday. That's what I'm saying. I can take it on Monday.
Oh, so Monday, make it on Monday then, because it's going to be too. What the other group said, Monday is okay for you guys to review and study.
Yes,
Yes,
Miss.
please, Monday.
Yeah, I think so, because it's going to be, because you have to go through, like you guys did very good when you got the reviews and then you work on the weekends. So yeah, so then Friday maybe you are going to work on the reviews also. You guys can study, you can post it, and then you can work on that weekend, and Monday you can set the exam. Okay.
Okay, thank you.
Okay, so we are going to be done soon. You guys can go now. It's already 12:21. Thank you for listening. Okay. Okay.
Thank you. Have a good
Bye now.
afternoon.
You guys too. Okay. Okay.
Good afternoon. Bye.
Okay. Bye bye
Okay,
now.
Thank you so
Bye.
much. Miss. Go sleep. Get some rest.
Yeah, you have to sleep. I have the night shift again tonight.
Oh,
Yeah.
wow.
Yeah.
You're strong.
Okay. 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 Thank you.
Okay. Bye.
Bye bye.
Bye. Thank you.
Bye.